Abhinandan. Good afternoon, everybody. Welcome to the one-day national webinar on severe antioxidants, chemistry, biology, and uses. But sorry for little delay. Please bear with us. We will start our webinar within few minutes. So please stay with us. We will provide the YouTube link in the WhatsApp group very shortly. I am Riddhi Ghosh, faculty of the Department of Botany, Kalna College, hosting the webinar organized by the Department of Botany, collaboration with the IQSC Kalna College, Kalna, Purbo Bardhaman. The last 1.5 years, our world has changed drastically. We have taken giant steps into future and made advancements that was probably due some 30 years down the line. Our world has shifted from the outside and is now totally confined not only indoors but also within the limited space of our electronic device. As is the saying, the world will go on, and it is only the fittest who can adapt will survive. Our survival today attests the fact that we have definitely adapted. Before I pass it to our Honorable Principal Sir, Dr. Tapush Samanto, for his welcome address to start the webinar, I humbly request all the participants to keep your microphone and camera in mute mode. Please do not share your screen. দয়া করে প্রত্যেকে আপনারা আপনাদের স্ক্রিনের মাইক্রোফোন এবং ক্যামেরা অপশনটি ওয়েবিনারের সুষ্ঠু পরিচালনার স্বার্থে বন্ধ রাখবেন এবং স্ক্রিন প্রেজেন্টেশনের অপশনটা অন করবেন না। I repeat, please keep your audio mute and video off in the webinar. Please do not share your screen. For your kind information, put your queries related to the talks using live chat box, and relevant questions will be passed to our speaker by us. We will forward you a feedback link after the question answer session of our webinar. Participants will get the certificate within 10 working days. Now, it is over to our respected principal, sir. Sir, please speak something about today's webinar.
Good afternoon, everyone. There is same technical issue as due to that our principal sir is now unable to join. We have to wait for some time, or Professor Somen Bhattacharya, if you are ready to deliver your speech, so I would like to request Riddhi ma'am to introduce our honorable speaker. Our principal sir will join later on.
That will be better if start now.
Yes, sir. Yes, sir.
At the time.
Riddhi ma'am, please introduce our speaker.
Okay, sir. Okay, sir. Now it is my honor to introduce our speaker of the day, Professor Somen Bhattacharya sir, Professor and Coordinator UGC Center for Advanced Study, Department of Botany, University of Bardwan, West Bengal, India. Currently, he is working on promotion of underutilized medicinal plants with an objective to promote and domesticate some medicinally important plants for further consultancy and marketing. He is also pursuing his research on crop diversification with amaranths as the most promising underutilized crops for their phytochemicals with antioxidants potential to combat degenerative disease. Formerly, he was a faculty of the Department of Botany in Hooghly Motion College, Krishnanagar Government College under WBES, and then he served Delhi University Constituent College. He was also a senior scientist selected by Agriculture Scientist Recruitment Board of Indian Council of Agricultural Research, Government of India, and posted at Vivekananda Institute of Hill Agriculture, Uttarakhand. He has 24 years of teaching experience and 26 years of research experience. His research interest includes plant redox biology, trace signaling, oxidative stress, and yield potential of crops, phytochemistry, and antioxidants. Some of his research work includes characterization of redox regulatory mechanism during germination of rice under abiotic stress, characterization of co-inducibility of trace proteins with central role of ROS as elicitor, understanding the implications of the role of calcium in the management of oxidative stress, and many more. He is a member of many prestigious societies, including European Society of Free Radical Biology, Plant Physiology Forum, etc. Presently, he has 7 research scholars and his 4 scholars already completed PhD and number of lectures in several national and international platforms. Many of his students are serving as faculty in several colleges throughout the country. Till date, he has published more than 100 research papers in many reputed international and peer-reviewed journals. His total citation is more than 1500 articles in national and international journals. With this brief introduction, I request Professor Bhattacharya sir to deliver his presentation entitled Severe Antioxidants, Chemistry, Biology and Uses. Sir, please.
At the outset, I would like to acknowledge my thanks and regards to the organizer for giving me a platform to share some of my work which I have done, or some of my studies which is relevant to this particular topic. And I would also like to acknowledge thanks to the principal of this college, Kalna College, for giving me the opportunity to share my thoughts or to share my ideas related to this very important topic. Now, with this, I also thank Miss Riddhi Ghosh, Madam, rather, who took all the strength to introduce me with so many things, so many, objectives and others. And I also thank Jaideep for being so kind to me to allow me to say something anyway. And but preferably, I'm a student of plant redox biology. I have been pursuing this domain for more than 2.5 decades, but I learned very little. My understanding on redox biology, particularly the component antioxidant and reactive oxidant species, their interactions, so on and so forth, is very limited. But whatever I understood, I'll try to deliver in context to the chosen topic. So without wasting time, I'm going directly to the presentation. Please allow me. Is it visible on your screen?
Yes, sir.
Yes. Should I put it in power in this mode? Okay. I'll request everybody to, mute their microphone. Otherwise, there'll be resonance, and that will disturb the character of the sound. So, the topic that I've chosen to say something about, out of making some story out of it, is antioxidants is chemistry, biology, and function. Now, antioxidant is a very hot topic nowadays, because if you just go for Google search, you'll find that you have out of every 15 publication, you'll have one publication from redox system, where antioxidant is a very important part. So you can understand, cutting across the workers, plants, it may be plant science, workers from plant science, animal science, microbial science, biotechnology, agriculture, then animal science, human physiology. These particular domain redox biology is common. So naturally, the more work is going on, and the relevance of this topic is getting momentum because of another very important thing, that is due to climate change and other things, excessive pollution and population, whatever, which you couldn't manage in proper terms. The occurrence of degenerative diseases increasing in one hand, and so people are trying to combat this thing at the preventive stage. That's one thing, and another, an antioxidant is there to common people, or to common people. Antioxidant therapy is very relevant in that context. So antioxidant becoming very relevant in that way, in medical terms, or related to health issues. And so far as plant is concerned, there is a huge population pressure. At the same time, there is climate change induced environmental stresses. So, and that imposes a kind of adverse effect on plants' productivity. And if you see the reason of declining trend in plant productivity, one of the important declining trend is obviously, it's metabolic dysfunction. Or you'll be surprised to know that a plant can harvest only 1/4 of the yield potential of the crop. The rest are being lost. And if you try to dissect the reasons behind this particular episode of non-realization of yield potential of the crop, particularly in the era of climate change, you'll find that redox is a very important aspect, which actually contributes a huge in terms of both loss of productivity, as well as its relevance in imposing plant tolerance. So what I'm telling you is that redox biology, to be very precise, is very relevant nowadays. And antioxidant is a component of redox biology. We need to understand, purify our knowledge, because most of us is to understand antioxidant as some chemicals or something like that, which prevent oxidative damage, nothing else. And in that way, they help the organism to combat stress, to prevent aging, and to realize potential, to go for development properly, etc., etc. But it's not like that. At the end of the day, or at the end of this episode of my speech, at least I will try to make you understand the role of antioxidants is still an intriguing question in plant redox biology. So I used to say like this, understand the entire thing in this way, that antioxidant occupies central position in plant redox biology. If you see the redox biology as such, not only plant but animal redox biology or redox biology as such, there are, broadly speaking, there are 6 components that are very important, that constitute redox biology, or the redox system of the cell. One is reactive oxygen turnover of the reactive oxygen species, or the Ross turnover dynamics. Ross stands for reactive oxygen species. I'll come what it is in detail. And the second one is its counterpart. Whatever knowledge you are having, we know that these ROS and antioxidants are just working in opposite fashion. So they are the contradictory. They are the agent which is to work on ROS, and they is to prevent their toxicity, or they is to quench them, they is to chew them up, whatever. So the second important component is antioxidative defense, and third one is ROS sensing. And what it is, I'll tell you. And then ROS redox signaling. I'm not saying ROS signaling, I'm saying redox signaling, where antioxidant is equally an important part apart from ROS. And then the crosstalk with other bona fide signaling pathway so far as redox system and hormonal system is concerned. In you know that in nature, under in vivo system, if you it's not the particular. If you think that only auxins are working, gibberellins are working alone, or cytokinin is working, or a particular working is alone, as usual. This way we are used to think, but it's not like that. In a system, there is a synergism, and there is crosstalk with each and every signaling process. And in that system, redox occupies the central position. That they influence the different ways, both feed forward and feed backward with the hormonal systems, or the growth factor system, or growth signaling factor system. And ultimately, there's another component, that is the differential expression of the redox sensitive genes. Expression of redox sensitive genes is omitted over here, but please put it down. So there is 6 components that togetherly constitutes, and the flow of information. Actually, this is nothing but the flow of information, and the information is redox information. And to be very precise, what do you mean by redox information? Redox information means, there are in a cell, there are plethora of chemical reactions where there is reduction, oxidation, and the same molecule can undergo reduce, same molecule can undergo oxidized, whatsoever it may be. So and if you just for example, if you see one very important life process, that is the metabolic process associated with the formation of energy, that is ATP. If you try to go in detail about it, how it is happening, you will find this is nothing but a redox system, maybe photophosphorylation or oxidative phosphorylation. It is actually the redox system which makes it possible. Because these redox components are all integrated in the membrane, and they are asymmetric, and electron mobilization is there. And while electron mobilization is taking place, the proton gradient is established. Using that proton gradient, metabolic energy is generated. This is how it's happening. So basically, it is the redox things, and these redox reactions are the part of the metabolic events, the life-sustaining processes. So what's wrong in it? Because these redox systems are there, and in this context, let me tell you that I used to say in that way that the origin of molecular oxygen is one of the greatest paradoxes in the biological world. Why it is so? Because the very molecule which sustains the aerobic life, because before the origin of oxygen by oxygenic photosynthesis, earth was anoxic, and there was anaerobic respiration only. And if you compare the energy output of anaerobic and aerobic respiration, there's a huge, and probably this is one of the cause, the cellular diversification or multicellularity, which is an energy dependent process, couldn't happen. So with the origin of oxygen, there is a evolution, chemical evolution from anaerobic to aerobic respiration, and that affords the cell to have the greater energy output from the metabolites they respire, and an cell is experiencing greater magnitude of energy, and that helps them out all the way to diversify their life and life forms, and the evolution took place at a greater pace. So that's there. But the problem with oxygen is that this is the molecule. If you have to reduce oxygen, because it's very strong. If you see the aerobic respiration itself. Oxygen is in the terminal end of the entire ETC, and oxygen, nobody is having the greater affinity for the electron in that particular chain of events. So oxygen has the strongest thermodynamic tendency to receive the electron, and when they receive the electron, they receive electron one after another. And to reduce oxygen completely, you need to have 4 electrons. You need to have 4 electrons. You see, this cartoon shows that when oxygen molecule takes up one electron, it forms superoxide. It should have been 2, okay, and dot minus. And when they take another electron, it forms and 2 protons from the system. They produce hydrogen peroxide. Then another electron and proton to phantom reaction, they produce hydroxyl radical. And then from hydroxyl radical, another electron proton addition causes the complete reduction of oxygen producing water. So that's there. So now you can ask me that why not all the 4 electrons can be added at a time. That's not possible because of spin restriction of electrons of the oxygen molecule. So you have to add 1 electron at a time, and ultimately you are getting superoxide, hydrogen peroxide, hydroxyl radical, and then water. Now the problem is, if you go for incomplete reduction of oxygen, you come up with the formation of superoxide, 1 electron reduction product. And if it leaves from this chain of events, then it can do a lot of things, because it has 3 electron deficiency. So it became a strong oxidant. It can interact with anything else, any biomolecules for your example. It can work with it can snatch electron from proteins, thiol containing proteins. It can preferably it can snatch electron from lipids, membrane lipids. It can snatch electron from even the enzymes, the DNA, whatnot. And then if it forms hydrogen peroxide, still have 2 electron deficiency, and it's the same way it remains oxidant, moderately strong oxidant, and when it takes up another electron, it becomes hydroxyl radical, the most potent reactive oxygen species. Okay, and it can react with anything else, and particularly the nucleic acids, and cause depurination, membrane lipid peroxidation, many things. So all these things are very strong oxidant, and they're very reactive, and that's why we call them reactive oxygen species. So you cannot avoid formation of reactive oxygen species. So when the mother nature has selected oxygen, as a very important component of the aerobic life, and our life depends on oxygen for many reasons, because oxygen is the main molecule, which makes this universe conducible for living. Because had there been no oxygen, it wouldn't have been go to the upper troposphere, and there wouldn't have been, allotropic conversion between oxygen and ozone, and our life wouldn't have been safe from ultraviolet radiation. Because when oxygen and ozone allotropic conversion takes place, they consumes the lethal ultraviolet radiation, and that's why the life being able to migrate from water to land. So all these things happen. So and now the tricky problem lies with oxygen in the sense that if there is incomplete reduction, I'm saying if, underline if there is incomplete reduction, that creates a hell of a problem, because you are going to produce many things which are very toxic, strong oxidant, and they can interact with any biomolecules. So biomolecules are at risk. That's the problem. Now, in order to counter this, cell produce antioxidant, and you will be surprised to know that the antioxidant, particularly the antioxidative enzymes, happens to be the constant companion of the oxygen, still its origin. So whenever you have oxygen-based life, oxygen is being selected as a very important parameter, very important component of aerobic respiration, and for realization of as much as free energy is possible from the metabolite that they oxidize. In that time onward, they know that this kind of event could have happened, and for that they produce antioxidants. Now, if you see these cartoons simply I have given to make you, because you cannot understand anything unless about antioxidant biology unless and until you know something about ROS. Because reactive, just now I told you these are the important reactive oxygen species. 3 reactive oxygen species I talked about. Of these, the superoxide and the hydroxyl radical are free radicals, and H2O2 is obviously
not a free radical, because in order to be a free radical, there has to be an unpaired electron in the outermost orbital. So in that sense, H2O2 has 2 electrons, so it's not the unpaired. So that's why we cannot call it a free radical, but they are still reactive and strong oxidant. Now, these cartoons shows the subcellular local of ROS formation, because the ROS formation in the subcellular local is associated with its counteracting agent, that is the antioxidant. You get antioxidants were at the very side. This is a very fascinating thing to study, that whenever we study chloroplast, generation of ROS, or the say peroxisomal generation of the ROS in terms of the greater kinetics, if you compare the kinetics of the generation of the ROS, I mean, the ability to generate reactive oxygen species in plant cell, in which subcellular component that's maximum, you'll find that it is on, I mean, during daytime when there's light, it is the peroxisome which generates maximum oxygen. And there are 2 pathways in peroxisome which can generate reactive oxygen species like hydrogen peroxide. One is C2 pathway, you know it very well. This is a glycolate metabolism, or photorespiratory metabolism, where glycolate is being converted to glyoxylate. And another is xanthine to allotonin formation, where through uric acid it produces hydrogen peroxide. And if you in contrast, in chloroplast, there is zero cyclic electron flow that operates during redox, loss of redox homeostasis. In absence of NADP+, ferredoxin donates the electron to oxygen and superoxide and then H2O2, all this thing. So here, and if you see the genesis, I mean, this is 4030 millimole per meter square per second, I mean, the kinetics, and this is 10,000 nanomole per meter square per second. These are the 2 major contributor of reactive oxygen species. And surprisingly, since we are discussing on antioxidants, you'll be surprised to know, and it is true, that if you compare the abundance of the enzymatic as well as enzymatic abundance particularly, and the ROS quenchers, you'll find that that is maximum in chloroplast, followed by the peroxisome. So best, I mean, it's not an arbitrary thing. That's why cell is so intelligent. You see that they have kept the sentries which can take care of the ugly customer reactive oxygen species if they are being produced in the fast kinetics. So they have kept it in the subcellular local, ideally. So they accumulate it. Why? Because this is not good at all. If you allow them to build their concentration, this is not good at all. So this can create a lot of problem. So you should detoxify them, and for that there is antioxidants. I'll tell you what antioxidants do. Do all this thing. Well, I mean, obviously there are other things, but basically they take care of they take care of reactive oxygen species, so that the enhancement of the concentration of the reactive oxygen species cannot take place beyond the threshold value. Because had it been like that, then being a strong oxidant, they can attack any biomolecule, maybe functional, non-functional, architectural, whatsoever it may be. And in doing so, they do a catastrophic, kind of things inside the cell, having a lot of damages we call oxidative damages, that and that culminates in even the loss of functional life of the cell. And another important site of generation of ROS is the apoplast in the cell membrane, where NADPH oxidase, what we called as RBOH, respiratory burst oxidative homolog. There's an enzyme which is present, a membrane-bound enzyme facing apoplast, and they're taking oxygen and producing superoxide and then subsequently H2O2. And this H2O2, is a very important customer. Nowadays, we know its role is getting purified. Not only it has an evil role to play, but it can work as a sensor. That is, environmental sensor that you'll come to know. But what will be the concentration of the H2O, which is being managed by the antioxidants over here? Because from superoxide to H2O2, SOD is there, and from H2O2 to whether it will generate hydroxyl radical or not, that will be. That depends on the availability of peroxidase system or catalyst system. Okay, so that's another thing. Another very important site is the mitochondria in plant cell as well as in animal cell, where you can see that the ubiquinone and ubiquinone complex 3 transition can generate reactive oxygen species. Okay, this is the most important site where the UV quinone complex 3 transition. That is, if there is by any means there is a redox imbalance and the cytochrome B, iron cytochrome B system, this complex 3 is saturated with electron, then UV quinone, actually the UV quinol can directly give the electron to oxygen, producing superoxide and hydrogen peroxide. But the kinetics is too low compared to mitochondria's chloroplast. So in chloroplast during daytime, you can see the 4030 millimole per meter square per second is the kinetics of the generation of reactive oxygen species. Whereas in mitochondria, it is less than 216 nanomole. So far as plant cell is concerned. So that's the really chloroplast is the major site, followed by peroxisome, then the apoplastic NADPH oxidase, and then it is your mitochondria. Now there is another site which is called membrane lipid. Membrane lipid gets peroxidized. I'll show you how it's happening. And they are the major sites of the generation of the reactive oxygen species like singlet oxygen. In singlet oxygen is a reactive oxygen species, where the electron moves in the reverse direction. Outermost electron in the oxygen atom moves in the reverse direction. And they also produce membrane lipid peroxidation, also produce alkoxy radical, peroxy radical. The secondary radical species, okay, so they are again very toxic, so they can induce even apoptosis. So, and this is lipid peroxidation. I'm not going about that, but keep it in your mind that lipid peroxidation. Just now I told you this thing, that is how the membrane lipid is getting peroxidized. Okay, there are 3 different steps, and the lipid is ultimately producing different kinds of lipid radicals. That is peroxy radical, alkoxy radical, etc. And from these, epoxide, hydroperoxide, aldehyde, and the glycols are the products, ultimate product. And this is kind of a vitiating cycle, I used to say, because once lipid peroxidation starts, they generate a lot of reactive oxygen species like alkoxy peroxy radical, like this is the alkoxy radical, this is the peroxy radical, etc. And they again induce lipid peroxidation. So a lipid peroxidation, once it is induced, generates further radical species or reactive oxygen species, which further produces reactive oxygen species. So the basic reactive oxygen species in cell types, maybe plant and animal, are these, like superoxide, and I have given the cellular location and their interaction capacities. You can see, if it is a superoxide, they can interact generally with protein and hardly with lipid carbohydrate, and there is no evidence of its interaction with DNA. So superoxide the DNA is safe from superoxide in that way. Hydrogen peroxide, if you see, they can react to the protein, particularly sulfhydryl containing hydrogen, and proteins are a real target for the hydrogen peroxide. Now hydroxyl radical, they attack everything: nucleic acid, protein, and carbohydrate, lipid. So that's why we say hydroxyl radical is the most potent reactive oxygen species. Then the singlet oxygen, and they can also target the guanine of the nitrogenous base of the DNA, and some of the amino acids, not all that is associated with the protein, and the polyunsaturated fatty acid. In that way, alkoxy radical, peroxy radical, you can see they can target the proteins and the lipid, particularly the fatty acid, polyunsaturated fatty acid. So what is the implication? This thing is better, you can see. So what is the implication? So I told you that the reactive oxygen species is natural product. I can claim that all these things are a natural product of the cell, nothing to be worried about, because you are doing it, you are playing with the molecule which affords the cell with a greater abundance of the energy. So you have to take some risk, and the risk is incomplete reduction. Risk is their incomplete reduction, and when they suffer incomplete reduction, they produce kind of this kind of radical species like superoxide, hydroxyl radical, hydrogen peroxide, and then singlet oxygens. Okay, got the point. So, and if their production is aggravated, there can be 2 different kind of situation where the antioxidant, when you have this thing generated, antioxidant come into action. So it is just like a interaction in the metabolic phase, where ROS and antioxidant interacts. Under normal condition, when the situation is normal, I mean, where the antioxidant capacity is competent enough to take care of the reactive oxygen species that is being formed and keep the reactive oxygen species behind their threshold concentration. By different means, I haven't talked about how the reactive antioxidant works on the reactive oxygen species. That is their biology. I will tell you later on. Okay, so for the time being, assume that antioxidant can counter reactive oxygen species, and if the reactive oxygen species is an evil agent, for the time being, you assume that, but it's not like that. And then if they're balanced, perfectly balanced, so it is a homeostatic condition, and the system is maintaining its redox homeostasis, and the metabolic homeostasis is done, and there is the plant is the organism is under defensed vitality, or you can say in that way that everything is fine and normal. But if there is a tilt, here it seems there is a major tilt. That is, the ROS generation, the kinetics of the generation of the ROS is comparatively significantly more compared to the antioxidant capacity to scavenge them. If it is so, then the redox homeostasis of the system is disturbed towards pro-oxidant, towards accumulation of the pro-oxidant, and this condition is known as oxidative stress. This condition is known as oxidative stress, where it's the kind of a situation where the redox homeostasis of the cell is being disturbed towards the pro-oxidant, accumulation of the pro-oxidant, and that causes through their oxidative damages, that causes plethora of oxidative as you can say, they can attack any biomolecule, and and oxidative damage is an imminent factor. So this is an oxidative stress, you can say, and under these oxidative stress, many things can happen. This ROS can instigate programmed cell death, okay, and growth reduction or yield loss is the factor, and then they can cause molecular damage. Everything is under attack, you can see, because I have already told you that this ROS has the deficiency of electron in their outermost orbital, so they want to snatch electron from any kind of molecule that they can interact with, obviously owing the redox norms, and fulfill their electron, and the molecule from which they snatched the electron, they got oxidized. And if it is a structural molecule, if it is a functional molecule, then they cannot work properly. That's the point. And this thing is being manifested in terms of the cellular damage. That is, if your membrane lipid is damaged, obviously the membrane cannot maintain its integrity. So membrane cannot maintain its integrity in the sense that the molecular trafficking across the membrane and cannot be regulated properly. So that will create a huge problem, because there may be ion loss, there may be pH abnormalities, so on and so forth. Okay, so if the protein is being lost, if the proteins are the enzymatic protein, so that metabolism will be jeopardized. If the nucleic acid is depurinated, and if it is a functional gene, so naturally there will be mutation or whatsoever. And if the pigment is being attacked, naturally the role of the pigment in terms of scavenging solar energy or the photomorphogenetic pigments whatsoever it may be, they fail to function properly, and all this thing is being percolated down in their putative metabolic and defense function. So ultimately that will increase. If the oxidative stress works randomly, then we say the wear and tear will be high and the system is aging fast. But if oxidative stress is working very systematically and in a programmed manner, we can say that oxidative stress is now instigating senescence. That is genetically regulated termination of functional life of a cell or organ or whatever. So I hope you understood for the time being. Keep it in mind that, well, antioxidants are something which interacts with the reactive oxygen species at its metabolic interface and determine the redox status of the cell, made with the homeostatic condition. Or if there is in earlier picture, I have shown that if there is a minor tilt, there's a difference. This picture doesn't make difference. They have shown only the tilt. Okay, but if the tilt is a minor tilt, there may be difference whether the tilt, that is the accumulation of the ROS, is minor, or the accumulation ROS of this is the maximum. If there's a minor tilt or major tilt, the implications are different. If there's a major tilt, and naturally there will be huge accumulation of the reactive oxygen species, and the reactive oxygen species can work indiscriminately with all the cellular metabolites, causing oxidative stress and oxidative threat to the biomolecule. Everything is happening, whatever I showed. So oxidative modification of cellular components, impaired metabolism, metabolic dysfunction, impaired translocation of metabolites, whatnot, impaired photosynthesis, impaired life processes. And that causes, in case of the plant, that causes decline in the crop productivity. Whereas in case of the animals, like human being, it causes aging, degenerative disease. Okay, aging, and you'll be surprised to know more than 100 degenerative diseases. The etiology of more than 100 degenerative diseases in one way or another are connected with oxidative stress, either as a propagator or as a mediator. Okay, that I'll come to this aspect also. So if there's a major tilt, that means the production of ROS is too high compared to the ability of antioxidant to take care of it. So that really creates an oxidative stress, and oxidative stress become threat for the biomolecules, and they cause all these havocs to the metabolic system. And the implication is, so far plant is concerned, that will be metabolic dysfunction and loss of crop productivity. Whereas in animal, they can cause degenerative disease, aging, accelerates aging, and even death of the system. Okay, but if there's a minor tilt, that's the thing. So antioxidant is trying to buffer reactive oxygen species. I'm saying buffer. So the new term I'm just intentionally I've just used. The antioxidants as a redox buffer, where there is a slight tilt in the accumulation of the ROS, and the endogenous concentration of the ROS is being within, not as I say in the threshold zone, but just cross the threshold zone. Now these reactive oxygen species is used by the system as a redox signal. Okay, so this ROS antioxidant interaction, where there's a minor tilt minded, where antioxidants actually, I don't know actually what is happening. People says that it is antioxidant coupled redox buffering, that what will be the concentration of the ROS that will work there, and that can instigate a redox signaling or a cell signaling phenomena that is being controlled by the antioxidant. So antioxidant efficiency is a regulated phenomena, and this is what the latest literature demands. Look, antioxidant, it is you can say that, well, if there is a surge in the genesis of reactive oxygen species, the demand for antioxidant is higher. So there will be more generation of antioxidant, okay, to take care of it done. But I'm saying, but that when you cannot, the philosophy of the cell is like that, you know that I cannot avoid the formation of the ROS. So why don't you use that particular ugly customer for your own benefit? And that is what exactly cell has done. At least there are a lot of examples I have seen in my experiment also, that here antioxidants works as a redox buffer, and they control the endogenous concentration of the reactive oxygen species to such zone where the reactive oxygen species can instigate a redox signaling or its own signaling mechanism. I'll come to that, because it has got all the signaling components, just like your two-component signaling system and an elaborate redox signaling cascades. And in the downstream, they are going to affect or cause differential expression of the genes, and through that they cause stress accumulation. So there is difference, you see. I cannot say simply that all the time there is a major tilt and there is an oxidative stress, and oxidative stress can instigate damage, and the damage can cause many things. In case of plant, they are the main concern so far as the realization of the yield potential is concerned, whereas in case of the animal, they are causing several degenerative disease, accelerates aging, so on and so forth. But at the same time, if antioxidant and ROS interaction, which is a very dynamic event, if it's a regulated one, that we do not know. that whether this antioxidant ROS interaction is a regulated one or not. So then what's antioxidant? What is an antioxidant? To be very precise, I am now bringing this term. Now, it's very difficult, rarely defined rather also. So one suggested definition is that, or I'm also saying like that a substance when present in low abundance compared with those of oxidizable substance significantly delay or prevent oxidation of that substrate. Now let me explain. Suppose vitamin C ascorbic acid is an antioxidant. Okay, in system it is present. Suppose the concentration is 10 nanomolar. Take an arbitrary example. Now in the system where reactive oxygen species is generated sparingly or in a greater magnitude, whatever, there is membrane lipid also, and the concentration of the membrane lipid is in terms of free fatty acid is say 100 nanomolar. So it is 10 times more than the concentration of ascorbic acid. Now, if the concentration, try to understand, reactive oxygen species can interact with both the free fatty acid as well as the ascorbic acid. Okay, now it is the ascorbic acid which is quenching reactive oxygen species fast, so that reactive oxygen species cannot oxidize your fatty acid. Okay, so in spite of the thing that the oxidizable substance are present in the greater abundance and the antioxidant is present in the lower abundance, doesn't matter. Antioxidant has got greater affinity to donate the electron or to quench your through different mechanisms that I will go right now. Okay, try to explain how it works. That's chemistry. And so it takes care of reactive oxygen species in spite of having the low abundance compared to the oxidizable substrate. So you can have the greater abundance of the oxidizable substrate, protein, fatty acid, nucleic acids, blah, blah, blah. But doesn't matter. If your antioxidant is there, even in the very low concentration, they can donate electron or they can detoxify your reactive oxygen species. So the reactive oxygen species is not able to interact with the oxidizable substance. In that way, they are preventing oxidation of oxidizable substrate. And that's why we call them antioxidants. Understood? Now, antioxidants, they are not reductants in that way because they can work in different ways also. This is not only the thing that they donate their electron to the reactive oxygen species. Suppose it is a hydrogen peroxide and there is an antioxidant like glutathione. Glutathione donates 2 electron to hydrogen peroxide, and hydrogen peroxide is reduced to water. Fine, done. It is not that easy. No, it's not like that. Okay, and this is the only way antioxidant works. No, it's not like that. Antioxidant can work at the level of the genesis of the ROS. Anything which can prevent the generation of the ROS, production of the ROS, can also be called as an antioxidant. So that's why they are not simply reductant. There can be a question, good question. People ask that why antioxidants? You see that antioxidant ultimately donates electron to the reactive oxygen species, so they are reducing reactive oxygen species. So why can't you call the antioxidant as reductant? And the answer is very simple. So look, there are other ways that antioxidant can evoke their action, physiological action, that I will tell you. But for the time being, you can imagine a situation where the genesis, if you cut the formation of reactive oxygen species by a mechanism, suppose there is a molecule which can prevent the generation of reactive oxygen species, obviously they are antioxidants. Okay, so that way also they work. Okay, so there are, the concept of the antioxidant has grown like anything. Say they offer different lines of defense, you will come to know. So antioxidant are basically the substance which is present in the low concentration compared to the oxidizable substance, significantly delay or prevent their oxidation. This is the most classical definition given by Haverland law. Given by Halliwell and Guthrie. It was Halliwell who coined this definition long back. Okay, so this definition is on, and you can but this is not an inclusive definition. That is what I mean. There are other ways antioxidant can work. So an antioxidant is a molecule that donates an electron to a rampaging ROS and neutralize it. That's fine, thus reducing its capacity to damage. these antioxidant delay and inhibit cellular damage. That's fine. And in that, if you put it in that context, you can call the antioxidants as a reductant. No problem. But since there are other ways antioxidant can work, just now I mentioned that antioxidants are not merely reductants, and they can prevent genesis of reactive oxygen species also. So that's why we cannot be satisfied with this definition. But at the end of the entire discussion, you can coin your own definition that will be much more inclusive, I hope. So there are some historical research on antioxidant, and it starts from the late 19th century or early 20th century, where mainly the antioxidants of industrial processes are being concerned. I mean, something when you add to the oil you can prevent the rancidity of the oil, oxidation of the oil. In that way, they have been used. But with the discovery of three important vitamins, vitamin A, C, and E, which obviously work as an antioxidant, that revolutionized the idea of redox system, particularly the antioxidant biochemistry of the living organism. So the broad idea about how antioxidant works, particularly in cellular system, in preventing aging, that was the work in in between 1930-40 to the 1960-70. So people worked in that way, that they used to call it as purely as an anti-aging factors. So if it is vitamin A, C, and E, people most of the people thought in that way, that they are nothing but anti-aging factor, because ROS is causing aging, causing oxidative stress, and oxidative stress imposes aging. So if you have these customers like vitamin A, C, E, which are having antioxidant properties, they can prevent better way, or they take care of this ROS and prevent the oxidative induced secondary oxidative stress, and that way they prevent aging. So it's an anti-aging factor. People used to think in that way. But the possible mechanism of action of the antioxidant was first explored when it was recognized that a substance with antioxidant activity is likely to be one that itself readily oxidized. So that's the very important thing, that how it's getting itself oxidized, that is it voluntarily donates its electron in a competitive redox environment. That's the point. So why an antioxidant voluntarily donates its electron in a competitive redox environment? Keep it in your mind and put it in with an interrogation. I'll try to address this issue later on. So plant, as I am from plant biology, people, and naturally my discussion will be a little bit biased towards the plant antioxidant research, though I will try my best to give some idea about the animal antioxidants and their implications in aging and etc. But the plant antioxidant research, if you track, it got momentum after the discovery of two very important antioxidant, non-enzymatic antioxidant and molecules, particularly, which is involved in redox regulation of photosynthesis, that is thioredoxin and glutaredoxin. And later on, it is being found that both thioredoxin and glutaredoxins are associated with redox regulation of photosynthesis, nitrogen metabolism, and other things. Okay. Now, I'll tell you a little bit how it works, but let me tell you that there are three basic mechanisms that antioxidant can evoke their action. One, their chain breaking mechanism, by which the primary antioxidants, I told you, donates an electron to the ROS in a competitive redox environment where there are a lot of oxidizable substance. Just now I told you, suppose there is an antioxidant A, and there is ROS, there is some ROS concentration. Okay. Now there is some other oxidizable substances also, which can be easily oxidized by ROS. So ROS can easily draw electron from, say, proteins, amino acids, lipids, so on and so forth. But since in that environment there are some antioxidants, though they are in low concentration, they donate electron in advance to the ROS and quench them, or rather reduce them to water. That's the point. So they are breaking the chain reaction in that sense. And the second mechanism involves the removal of ROS or reactive oxygen species initiator, that is secondary antioxidant, by quenching chain initiating catalyst. That means there are certain, suppose H2O, from H2O2, hydroxyl radical need to be formed. And it needs, I mean, there are some transition metal ion which is required. Now, there are some antioxidant which can chelate this transition metal ions, and in that way, these transition metal ions are not being available to the system. So, H2O2 to hydroxyl radical formation is being stunted or is being blocked. So, this is how it works. Clear? And there may be some other ways that antioxidant may exert their biological system, effect in the biological system as co-antioxidants, that is helping antioxidants, and through gene regulation. Underline this, through expression of gene regulation. You will be surprised to know, I'll show you that how antioxidant can cause its own generation through de novo expression of genes. So in that way, suppose you have got some antioxidants whose concentration is very meager in the cell, and now cell is encountered with a stress, maybe biotic or abiotic, and you need to defend yourself. Now, how can you do this? This is the question. So you can do this very well. I mean, now it is being explored that, well, here antioxidant ROS interacts in a very moderate way and in a very regulated way, generating a certain concentration of ROS, which is being used or instigated as the defenses, as the redox signal, and they use different redox signaling cascade, and they ultimately activates the transcription factors of antioxidative genes, producing its own synthesis. Okay, so this is how it can work. So antioxidants can itself, through their redox regulation property, can influence expression of their own genes. In that way, they increase or enhance their own production. Now, I used to prefer, as to put it in that way, that there are 4 lines of defense that antioxidant can give us, may it be plants or animals or microbes. The first line of defense antioxidant molecules we call preventive antioxidants. Here, the preventive antioxidants suppress the formation of free radicals. Okay, that is, antioxidant reduce hydroperoxides and hydrogen peroxide like things, beforehand to alcohols and water, respectively, without generation of further free radicals. And we know that from hydrogen peroxide, hydroxyl radical can be formed. From hydroperoxide, singlet oxygen can be formed. So if you can, these are the antioxidant which take care of these things, which is the initiator or the product, which is the rather you can say, which is the initiator of the formation of other potent reactive oxygen species. So in that way, they work. That means they prevent the formation of the potent reactive oxygen species by interacting with the moderate and reactive oxygen species like hydroperoxide, hydrogen peroxide, and convert them into water or alcohol, so that they are not in a position to produce the most potent reactive oxygen species. Okay, and there may be some proteins or secondary metabolite sequestering agents also. Okay, so you can put it in that way. And glutathione peroxidase, glutathione ACE transferase, and the phospholipid hydroperoxide glutathione peroxidase and peroxidase are known to decompose lipid hydroperoxides. Otherwise, these lipid hydroperoxide would have given you some other secondary toxic, very toxic reactive oxygen species like lipid radical, alkoxy radical, peroxy radical, so on and so forth, and that could have create a lot of problems so far as imposing oxidative stress is concerned. So this is the first line of defense, that is a preventive antioxidant. Second line of defense is called the scavenging antioxidant. We already know that just now I told you there's a chain breaking reaction that they themselves give the their own electrons. Here the antioxidants scavenge the active radicals to suppress the chain reaction, break the chain reaction, propagation reaction particularly. Okay, so that means they simply donate their electrons. Okay, so they are, in this case, they are simply the reductant, you can say. So they can vitamin C, uric acid, bilirubin, albumin, thiols, this is the hydrophilic radicals, scavenging antioxidant, vitamin E, ubiquinol, all these are of this category that they donate the electron. Suppose you have hydrogen peroxide formed and there is scope of formation of hydroxyl radical. Suppose you have got the superoxide form and you have the scope of formation of hydrogen peroxide hydroxyl radical. Suppose you have formed the lipid radicals and you have the scope of formation of alkoxy radical, peroxy radical, singlet oxy, and so on and so forth. So here this chain reaction could generate many more reactive oxygen species, and the antioxidant work at the first level. By donating the electron they need, they reduce them completely. So if it is superoxide, they donate the electron with 3 electrons rather, and the superoxide is reduced to water one after another. They give the 3 electrons, superoxide to H2O2, H2O2 to hydroxyl radical to water, done. Okay, if it is hydroxyl radical, 1 electron, done. So they break the chain, and had the chain been not broken down, then they would have produced much more reactive oxygen species. So they are the scavenging antioxidant in that way, which breaks the chain by donating the electron. Okay, and so this is the second line of defense. Third line of defense is the repair antioxidants. So here the antioxidant, these are the repair de novo antioxidant, and the proteolytic enzymes like proteases, peptidases, which are present in different subcellular organelles of the cytosol, they remove oxidatively modified proteins and prevent the accumulation of oxidized proteins. So you know that if you have, for example, if you have a partially degraded protein, okay, or oxidized protein, and you form if the protein got oxidized, you are getting your cell will be accumulated with free carbonyl compounds. So, and if you allow them, they can further produce some toxic events. Okay, for example, during glycolysis, methyl glyoxal can form. Methyl glyoxal can be produced, and if you allow the methyl glyoxal to be accumulated, then it can cause toxicity to the system. Now you have to cells develop 2 important enzymes, methyl glyoxal is 1 and 2, which take care of methyl glyoxal and degrade them. And this is how it's happening. So the third line of defense is the repair antioxidant. That is, here you are repairing the damage. So damage is being done, and if the damage is being accumulated, that would have caused much more cost to the cell. And here your antioxidant defense system come into action and rescue your cell from that. So that's how it works. It's wonderfully done. And the DNA repair mechanism is another kind of example you can cite, where the DNA repair system also play an important role in total defense system under oxidative stress, because you know DNA is not safe from oxidative stress. Particularly hydroxyl radical, they are very prudent to attack the nucleic acid, causing depurination and etc. And this can be, and if it is done, these damaged product need to be taken care of. Otherwise, they can produce many things, okay, which can be much more toxic. So that's how it's being done. So repair mechanism is a very important thing, particularly repair against oxidatively damaged metabolite. And here lies the significance of antioxidant. And last not the least is antioxidant coupled redox regulation. Just now I told you where the role of antioxidant is very fascinating. Here, as I told you, the antioxidant works as a redox buffer. They regulate the concentration of ROS and they generate, rather sometimes they allow the generation of the ROS intentionally, so that the ROS concentration can be built up to a threshold level. For example, I'm telling you an example, that whenever you have an infection in a biotrophic cell, in a biotrophic host, suppose a pathogen infects a biotrophic host, at the level of the leaf, or the epidermal infection is there. Now, immediately, cell using their sensors, they activate NADPH oxidase, producing H2O2. How they are producing H2O2? Because NADPH oxidase, they produce superoxide. And from superoxide, H2O2 can be produced. Now, superoxide to H2O2, which enzyme is required? Superoxide dismutase. But there are other enzymes as well. What are those enzymes? now H2O2 can be broken down to, or rather H2O2 can be converted to hydroxyl radical, and hydroxyl radical to your water. But you see that they restrict the action of catalase and peroxidase. So under this situation, the activity, there's an intentional down regulation in the activity of antioxidative enzyme, catalase and peroxidase, allowing the H2O2 concentration to grow to a threshold concentration. Okay, so these H2O2 now work as a systemic signal. They migrate all the way from the epidermal cell to the inner cortical cell, and where they will induce the apoptotic genes, okay, or causing programmed cell death. By the time your pathogen invades the epidermal cells and come to those cells, cortical cells already got sacrificed their life, and your pathogen is tracked by the dead cells. And this is hypersensitive reaction, you know very well. So here, what was not known previously, people thought that it is salicylic acid which is to do all the trick. But no, it is actually, though salicylic acid is the customer, but the main thing is done by the Ross wave. Here H2O2 work as a Ross wave, and they migrate from one cell to another cell through through the apoplast, and they go to the inner cortical cell, where they induce the apoptotic genes and the programmed cell death genes, and they sacrifice their cell in advance. By the time pathogen enters there, they are wrapped by the dead cells, and there is no way they can survive. So here you can see that antioxidant, it is called antioxidant coupled redox regulation. Why? And you know, because here antioxidant activity is being modulated, it is regulated, and towards such a magnitude that it can give you a redox signal, and the cell can exploit that redox signal. Using that redox signal, they can induce specific genes required for a developmental process. And here senescence is the developmental process. You all know it's the terminal developmental process. So antioxidant, we call this an adaptive signaling antioxidant. So now you apply your sense. Previously, what people used to think of that antioxidant, well, this is a customer, we just interact with Ross, reduce their concentration. But no, sir, this is not done. This is not the right thing. There are many things associated with antioxidant biological function. Okay, now you come to the diversification of antioxidant, because there are many things that is inherent with the diversification of antioxidant function. Basically, we are having non-enzymatic and enzymatic antioxidants, as you all know. Probably, and enzymatic antioxidants are basically depending on ascorbate glutathione pool, and there are 4 enzymes: ascorbate peroxidase, glutathione monodehydroascorbate reductase, dehydroascorbate reductase, and glutathione reductase. So all these enzyme activity and their antioxidant potential depends on ascorbate glutathione pool of the system, and others are superoxide dismutase, catalase, peroxidase, polyphenol oxidase, glutathione peroxidase, glycol peroxidase, glutathionase transferase, peroxiredoxin, thioredoxin, etc. Okay, so these are all enzymatic antioxidants. Okay, so one class of enzymatic antioxidant depends on ascorbic acid and glutathione, because they are the components which actually donates the electron, and they are the most important redox customer that is behind the curtain, behind the activity of this enzyme. So keep it in that way. And these are others which doesn't depend on the pool of any other redox agents. Okay, they work of their own. Superoxide dismutase, you know very well, it works on superoxide producing H2O2. Catalase, it works on H2O2. Without the assistance of other cofactors, detoxifying it to water. Peroxidase, with the help of a co-reactant, that may be glutathione, that may be glycol, etc., etc., that produces water, or that detoxify H2O2. Then polyphenol oxidase, that's a very important Enzyme also similarly, glycol peroxidase, glutathione transferase, peroxidoxin, thyrodoxin. These are all involved in detoxification of the reactive oxygen species in their own way. And so far as non-enzymatic antioxidants are concerned, obviously ascorbate glutathione is the most important one, as I told you, because they instigates the activity of these enzymes, which operates in tandem to give a pathway, what we call as ascorbate glutathione pathway. I'll tell it later on. And then you have some non-enzymatic compounds, or low molecular weight compounds like tocopherol, carotenoids, flavonoids, alkalic acids, non-protein amino acids, so on and so forth. So all these things, even proline can be a very good soluble sugars. They are very good antioxidants. The modern literature says that these osmolytes, particularly proline and soluble sugar, are very good antioxidants. So they perform dual role in the system. And antioxidants work quickly, and this is an ideal example where you have seen that this hydrogen peroxide can be generated by different ways. Through superoxide dismutation, and superoxide can be generated, for example, in plant cell, I have shown you, through pseudocyclic electron flow from ferredoxin. Okay, and superoxide can be dismutated to form H2O2, and H2O2 being the most stable reactive oxygen species with half-life almost 2 milliseconds, they're going to stay for a longer duration, and they can evoke oxidative threat to the system. Because there is a concept called because H2O2 itself can activate many NADPH oxidase, producing many superoxide. Okay, so we say microbar. So once say few H2O2 molecule is formed, this H2O2 molecule is able to generate several thousands of H2O2 within few minutes, through NADPH oxidase or respiratory burst oxidative homolog, which is a membrane-bound enzyme. So H2O2 is a very serious customer oxidant within the cell reactive oxygen species. They need to be taken care of, and they are taken care of by ascorbate glutathione system, where you can see that H2O2 is reduced by ascorbic acid donating the electron to H2O2 2 electron, and the enzyme involved is ascorbate peroxidase, and you are getting water done. Your job of detoxification of H2O2 is done. But when ascorbic acid donates the electron, it being converted into monodihydroascorbate. It's an oxidized form, and in order to keep the cycle on, you have to again get it converted to ascorbic acid, and that can be done with the help of NADH. That is the reducing power, not NADH, reducing power generated by the jet skimmer, your pentose phosphate pathway, with the help of the enzyme monodihydroascorbate reductase directly, and you got ascorbic acid back. Sometime monodihydroascorbic acid is getting converted into dihydroascorbate, and dihydroascorbate is then further reduced by glutathione with the help of dihydroascorbate reductase, and you are getting ascorbic acid back. But the glutathione is what is glutathione? It is a tripeptide, which is nothing but the gamma glutamyl cysteine glycine. There are two peptide bonds in between, and this glutathione, having the sulfitryl group, donates the electron to dihydroascorbate, in fact, and glutathione is converted into oxidized glutathione. So again, oxidized glutathione has to be converted into reduced glutathione to keep the cycle on, and it is the NADPH, which is the product of pentose phosphate pathway or jet schema photosynthesis. They donate the electron finally, and with the help of glutathione reductase, glutathione is back to the system. And the entire cycle is, deduced by Halliwell and Asada, and we call them Halliwell-Asada pathway, or you can safely call them ascorbate glutathione pathway. Okay, and if the cell is being exposed to xenobiotics, that is the toxins, then there is again the role of glutathione to detoxify them, because this is the glutathione. Then glutathione, with the help of the glutathionase transferase, they are reducing them. Okay, they are reducing them. And not only that, they're putting them inside the transportable vacuole. Okay, and the glutathione can also be, this glutathione, H2O2 and the alkoxy radical can again be detoxified by the glutathione glutathione S transferase, glutathione peroxidase or glutathione S transferase system. Okay, and they can be converted into glutathione, but this glutathione, oxidized glutathione, can again be utilized in the same reaction and to get your glutathione back. So the role of these antioxidants, diversified antioxidants, the take-home message is that you see that which antioxidant plant will recruit under which situation, it is again a very specific event. It is not like that, say, sometimes they're using, when they're trying to manage photooxidative stress, which is developing under excess for solar energy, they're trying to, sometimes they're using ascorbate glutathione pathway, sometimes they're using glutaredoxin pathway, sometimes they're using secondary metabolite pathway. It's not like that. It is highly ordered, highly regulated, and as I told you, there are definitely antioxidant coupled redox regulation, and the genes are definitely involved in integrating or orchestrating the entire antioxidative events. So whatever I told you, it's being written over there. This is the antioxidative systems, and these are the targets with which they can interact with, and mostly where they are localized. I have given for example, Halleyville pathway, they can take care of H2O2, superoxide based H2O2 or H2O2 produced of the NADPH oxidase, and they are generally, in plant cell, they are operating in the chloroplast. Okay. Similarly, glutathione peroxidase can be operating, they can take care of H2O2 definitely, and chloroplast, cytoplasm, endoplasmic reticulum, mitochondria. So, there are different, as I told you, that the recruitment of the antioxidant in different subcellular local with a different objective is highly precise, and that entails us, well, it is not a chaotic process that, well, you produce antioxidant, whenever you get a reactive oxygen species, you quench them and detoxify them. It's not like that. It's not like that. It is highly regulated, highly coordinated, way of action they exhibit, and ultimately they try to maintain the redox homeostasis. These are the just skeleton I've shown you, that low molecular weight antioxidants like glutathione, beta carotene, flavonoids, and these are all secondary metabolites of the plant, you can see, and they are very, very important. And so far as their quenching ability with the reactive oxygen species is concerned, and their ability to quench the transition metal ions responsible for the formation of the most potent reactive oxygen species, that is hydroxyl radical, is concerned. I told you that here, if you have the advantage of flavonoids over, say, glutathione, is that the flavonoid, they can donate electron to a reactive oxygen species, detoxify them, fine, direct chain breaking reaction. Or flavonoid, they can be protective one. How? Or the preventive one. How? They can chelate a transition metal ion like copper or iron, okay, and doesn't allow them to facilitate the reaction where the hydrogen peroxide is converted to hydroxyl radical. They are preventing them. So they are preventing the formation of reactive oxygen species. In that way, they can also work. So that's why they are much, diversified way of action, and the plant intentionally keep them in definite subcellular local and under definite under severe or moderate environmental threats, by shifting the carbon budget of the entire metabolite chemistry, so that they can take care of the redox system and they can maintain redox homeostasis. So these are another important low molecular weight antioxidant working. Ascorbic acid, you all know how important it is. It is not only as the direct chain breaking antioxidant, but it works in tandem with other reactive oxidants, with other antioxidants like glutathione, and involving at least four enzymes like ascorbate peroxidase, glutathione reductase, monodehydroascorbate reductase, dehydroascorbate reductase. They can take care of oxidative damage. Okay, or reducing the level of ROS. And the vitamin E is a very important one, and their derivatives, and they are capable. You'll be surprised to know that these vitamin A and C, when works synergistically, they can prevent membrane damage like anything. Okay, so if you apply vitamin C or alone and try to see what is its impact on membrane damage, oxidative membrane damage in terms of lipid peroxidation, you'll have one kind of result. If you apply vitamin E, you'll have another kind of result. Now, if you apply them togetherly, you'll find the effect is synergistic action of vitamin C and vitamin E, or different kinds of vitamin E. They are really very. I mean, there is an upregulation, many times upregulation, and they can take care of the thing properly. And this is the ubiquinone and some diallyl sulfide compounds. They are also very good antioxidants. You know, that is plant or the animal cell precisely are using this thing. And now I'm coming to the plant redox biology and the role played by antioxidants. Now I'm coming to the role of the antioxidants. You know, the very important thing. Now, we all know that under abiotic and biotic stress, there will be a redox imbalance. Why this is so? Because of enhancement of the concentration of reactive oxygen species. And these are the reactive oxygen species that accumulates under severe environment, depending on the severity of the environmental stress, may it be the biotic or abiotic, I mean environmental or infectious. And there is an antioxidative defense that try to combat the situation, or rather deal with this situation through ROS antioxidant interaction in the metabolic interface. But if the balance is tilted more towards the accumulation of the ROS, then it ensues oxidative stress, and if it is so, it can have 4 important avenues of damage. Broadly speaking, there can be protein damage, there can be lipid damage, nucleic acid damage, and membrane damage. And if this damage are not being taken care of once again by the antioxidative defense molecules, then you may have, you can see that the antioxidant defense can work in this area also. Okay, and here also antioxidant works. So antioxidant works in 2 sites: one at the site of genesis, and another at the site of the evocation of their action. Okay, so if they're not competent enough, once again, then it causes aging and cell death. Okay, and all these things can be identified. You know, all these component, its changes, etc., can be identified nowadays easily through different proteomic approaches. I'm not going in detail about that. Now, this is broadly speaking what is happening here. Now, when you have the environmental conditions like ambient environmental condition, normal environmental condition, no threat, plant is used to have, plant is conducible environmental condition in terms of the temperature, humidity, light, so on and so forth. So under this situation, you will have the generation of reactive oxygen species true, and that is basal. Okay, and you have an elaborate antioxidative defense system, that is the enzymatic, low molecular weight, non-enzymatic, quenchers, scavengers, whatever I told you. There is an interaction at the metabolic interface, and the resultant is the physiological titer of the endogenous ROS, which cell can explode, or is not threatening to the in terms of the oxidative damage. Okay, so the remnant ROS can be sensed through ROS sensing mechanism, and there are different ROS sensing mechanisms. I'll show you where the antioxidant again play a very important role, and they instigate a signaling cascades, and the signaling cascade in the downstream using the MAP kinase pathway and other things. They recruit these transcription factors like MEV, KISAT, RAY, HSF, so on and so forth, and these are all known transcription factors which are working under stress. So the stress-induced transcription factors are redox sensitive, and their activation is being done by the redox signal, and the redox signal is generated. How redox signal is generated? Redox signal is generated due to a very controlled interaction between the ROS that is being generated under normal environmental condition with an elaborate antioxidative system. I'm saying this is a very controlled interaction. And that would have, why I'm saying this, try to understand, these antioxidants would have chewed off all the reactive oxygen species. I mean, they could have finished all the reactive oxygen species, detoxified them into water, but they didn't. They kept some residual reactive oxygen species as a physiological titer of reactive oxygen species, which is a signal which the cell is using as a signaling molecule. This is the point I like to make over here. So under this situation, your reactive oxygen species is working as a signaling agent, and it's not like an arbitrary thing that they will influence these molecule and that molecule of the signaling molecule. No, there are specific redox sensors, ROS sensors. I'll show you. We have a very elaborate ROS sensing mechanism that can be done through thiol containing or non-thiol containing or antioxidant coupled sensors. And after sensing, just like a hormone is being sensed by its receptor, the ROS is being sensed by its own receptor. Okay, and ultimately a signaling cascade is operating, and through that, transcription factors are upregulated, and these transcription factors are the transcription factors of redox sensitive genes. And this may be the antioxidative genes, this may be the morphogenetic genes, this may be the photosynthetic genes. And through this, you can have morphogenetic response like lateral root elongation, the adventitious root formation. There are many. Okay, then stress acclimation. So you can have the defense molecules which is being produced under stress through this antioxidant coupled redox regulation and physiological response. Now, when the interaction between the antioxidant and the environmental stress is incompetent, in the sense that the stress is severe. There is an ambient environmental condition, but here there is a stress, and the stress may be sublethal or stress may be severe. Now, when the stress is severe, the interaction between this becomes sometimes incompetent, but they cannot directly take care of this thing through their redox buffering mechanism. So there is decontrolled accumulation of ROS, and these causes the oxidative damage and cytotoxicity, which causes genotoxicity and cell death. So ROS may not sometime being able to control the stress effect, particularly when the magnitude and the duration of the stress is high. Then the antioxidant is not being able to coordinate the response, or rather buffer the concentration of ROS to such a level which can work as a signaling agent. That's the point I like to make. So, but there are sometimes under sublethal stress, then the antioxidant become very promising, and they work in the metabolic phase, and they keep the titer of the reactive oxygen species in a very regulated manner, or they regulate the endogenous concentration of ROS, and the ROS then through their own sensing mechanism that they have exploited it over here also under ambient condition, and their own signaling cascade, and their cross-talking with ability with other hormonal signaling, they impact the transcription factors. Like these are the emergency transcription factors, preferably I used to say sometime, because they're all induced under stress, and had they are being these transcription factors are being activated, then you have their target is the redox sensitive genes, those genes which can produce sufficient amount of antioxidants and maybe enzymes, maybe secondary metabolites, maybe non-enzymatic antioxidants. Then they can give you the morphogenesis which is required under stress. Like if you have a acute drought stress, you need to produce cuticularization, heavy deposition of secondary wall material, and the root here should be more, okay, so that it can have the greater access to water. I mean, this can be done through this entire mechanism. It is being proved, and they can regulate photosynthesis also. Not only that, nitrogen metabolism also can also be regulated. So you can have the physiological response towards stress accumulation. The concept of redox regulation is further expanded. I hope you are not getting bored, because I will take another 15 to 20 minutes to wrap up. Please allow me, otherwise the story will be incomplete. So in plants, the regulation, redox regulation phenomena is from ferredoxin of, those who are the students of the plant science, they know ferredoxin is a very important component of the jet scheme of photosynthesis. And ferredoxin generally donate the electron to NADP plus, okay, through FNK protein. But sometimes ferredoxin donates the protein to thyroidoxin, and the thyroidoxin is responsible for activating your PCRC enzymes, thereby they are regulating the photosynthesis. Okay, and not only that, thyroidoxin is the component which can produce a reducing equivalent in terms of malate, and the malate can be utilized in the different locale of the cell as the reducing agent, and where they can again produce thyroidoxin and take control all of these particular episodes of metabolic events like different metabolisms, energy yielding process, protein synthesis, and stress response. These are the broader view I'm talking about, So the concept of the redox regulation is now not, so far as the plant is concerned, is basically not restricted with the reactive oxygen species, but it is, that it is a thyroidoxin, and this thyroidoxin is the antioxidant. Okay, it's a bona fide antioxidant. How thyroidoxin, glutaredoxin pathway, they control the entire episode of nitrogen metabolism, protein metabolism, stress response, ground metabolism, energy output through respiration. Everything is under control of this redox system. So you can put it in that way also. So just I'd like to mention it over here. If you see the light operating system, this is the jet scheme operating, and they are producing ferredoxin is giving the electron to NADP. NADPH is produced fine, but it sometimes can produce ferredoxin reduced, can get the electron to thyroidoxin. You can see, okay, and thyroidoxin is getting reduced, and the reduced thyroidoxin then can affect the target enzymes or target transcription factors even. Okay, and they can cause the redox signaling, and this redox signaling causes the expression of CAP genes and antioxidant genes like ascorbate peroxidase. So this way it can work. Okay, so you can see that how the redox, jet scheme doesn't mean that you have to produce only NADPH, only ATP. Keep it in your mind. The bigger sense of the jet scheme in holistic term says it can produce a whole lot of compounds, particularly the redox equivalents, which can influence its own gene expression, which can influence the defense machinery, which can influence the time-to-time need of the cell, particularly the green cell, under environmental assault. Just to mention that I told you that antioxidant can be involved in sensing hydrogen peroxide. You can see that the antioxidant couple mechanism, definitely reactive oxygen species. When the cell explore reactive oxygen species as a signaling agent, I'm claiming naturally there has to be some receptors. Basically, thiol compounds are receptored, and this is how it works. I'm not going about that. Now, there are non-thiol compound, how it works, I'm not going about that. But there are some antioxidants which can helps sensing the reactive oxygen species in the entire episode of redox signaling. So here, what is being done, the redox buffering and the non-availability of H2O2 for the activation and the repression of the transcription factor. This is important. That is, they can change their affinity. They can change their affinity for the reactive oxygen species. Sometimes they can get themselves down-regulated, and when they get themselves down-regulated, the concentration of ROS become higher, and these ROS is required for signaling. Done? Okay. Now, sometimes, suppose ROS concentration is getting too high, but you have to keep them in such a concentration where it can work as a signaling agent, not as an oxidizing agent. Okay. Then this antioxidant comes into action, and they do all this trick. And this is a synoptic understanding that where not only the people used to say the ROS is the redox signal. No, to my understanding, the role of antioxidant is much more important, because antioxidants, they interact with the ROS and make the physiological titer of the reactive oxygen species, which can be explored by the H2 specific redox sensors, exploiting the MAP kinase module or other hydrogen peroxide sensitive modules like UDP glucose transferase KOD peptide module, and they ultimately activate the transcription factors, and through those transcription factors, the redox sensitive genes, and you can see a whole lot of the events. They can influence programmed cell death, metabolic modulation, stress acclimation, senescence, stomatal regulation, developmental programs, whatnot. So you can see the role of antioxidant is not restricted. The take-home message is that the role of antioxidant is not restricted to detoxify your reactive oxygen species, but in broader terms, it can involve in so many developmental and stress acclimation process. So this can be simply, there are some transgenic, I've given some examples, and these transgenics, when you put manganese SOD from yeast to the rice and you make a rice transgenic, they're better adapted, you can see they maintain a high level of SOD and ascorbic activity, and they are better adapted under stress, okay? And similarly, manganese SOD in the Pisum sativum, from Pisum sativum in the rice is resistant to dehydration stress. Copper zinc SOD from this to rice transgenic, they're resistant to oxidative stress. So these are some of the examples I have given to make you understand, well, look, these genes are involved, we all know, for stress tolerance, but these genes, how it works in the downstream, it doesn't necessarily mean that only reactive oxygen species is the customer which is doing all the trick. I'm not going into about all this thing. I'm coming to the animal science precisely a little bit, and then I'll wrap it up. Now, so far as the animal cell is concerned, just from the birth through aging, ultimately you are aged, and if you see the mechanism of action of the aging, basically it is taking place through oxidative events, and the mitochondria is the main thing, and the mitochondria generates ROS, and the antioxidative defense try to buffer them, and when they become decontrolled, that gradually causes oxidative stress, and that causes membrane alteration or lipid protein or damage, oxidation, cytoskeleton damage, accumulation of aberrant protein, and then there will be organelle dysfunction. And if it happens in between, not due to aging, but due to some other reasons, if there's a spar in reactive oxygen species formation, maybe due to xenobiotics and others, then you have got this kind of manifestation of these diseases. Like, you can have obesity, atherosclerosis, dementia, osteoporosis, and above all, cancer. Okay, so the implication of ROS antioxidant interaction is being heavily identified. I'll give you some example where you can see that how it is important, and then these are all the effect, okay, that used to happen, and with aging, these incidents are increasing. Okay, these incidents are increasing with aging, but it doesn't rule out the fact that if you have the degenerative diseases like these diseases, also the same thing is being mimicked. So the aging related changes can be preponed when you have got this kind of disease, particularly and the metastasis, atherosclerosis, osteoporosis. These are all degenerative diseases, okay? And where there is a decontrolled ROS antioxidant interaction, and that leads to the secondary oxidative stress, and that aggravates oxidative membrane damage, that aggravates the oxidative membrane metabolite damage. Now, you can see that the loss of redox homeostasis and aging can be deterred by antioxidants. Now, particularly these antioxidants are very important, which is found to deter it. So which way, whether you will be towards this aging-induced phenomena, or just now I have shown you where you will have the accumulation of all these things, or oxidative damages, or age-related diseases, degenerative diseases, or inflammation, or abnormal cell death, whatever. these are the all consequences of these ROS-induced, ROS antioxidant decontrolled interaction, I must say. But had there been controlled interaction, these would manifestation wouldn't have been there. But if you have these things, you can see that they manage. Then there is a controlled interaction because you have a ample of these substances which is regulating the endogenous concentration of this and not allowing these things to happen. Okay, and that's why there will be antioxidative defense, there will be longevity and healthiness. Okay, so the aging depends on the bioavailability of these classes of compounds like vitamins, polyphenolic compounds, low molecular weight antioxidants, and then some minerals having antioxidant properties, carotenoids, antioxidant cofactors, so on and so forth. So if you have ample of these things, there is a lesser chance of degenerative disease. Okay, some example of therapeutic application of glutathione in cardiovascular disease, degenerative disease like cardiovascular disease. If you apply glutathione, the risk of cardiovascular disease is reduced significantly. Total plasmic glutathione content is lower than cardiovascular disease patient. Okay, compared to the healthy subject. Similarly, in type 2 diabetes, if you treat it with the glutathione in a very systematic manner, obviously by monitoring the endogenous concentration of antioxidants, this can protect your pathologies associated with the diabetic nephropathy. Okay, these are the references, the people have done this. And in the cancer, the free radical generation is lowered when you have administered glutathione or given glutathione to the system. Okay, and so the reactive oxygen species accumulation involved in the neurological complication getting lowered, and 60% of the colon cancer patient expressed high level of glutathione, particularly in tumor tissue. So this is a wonderful finding. There are many findings. I have just chosen a few to make you understand. Look, how antioxidant can have a controlled interaction with reactive oxygen species, deferring your aging or degenerative diseases. So in polyphenolic compound nowadays attracted much interest because of its unique two-phasic, biphasic antioxidant property. One way, they are directly chain-breaking agents. Another way, they are preventing genesis of the reactive oxygen species. So their efficiency, so far as antioxidants are concerned, is much better as compared to the others. So polyphenols, if you apply them in cancerous patient, there is a significant reduction in the risk of colorectal cancer. And there are ample of evidences, recent evidences are also there. Now, if you apply in the cardiovascular diseases, the risk of cardiovascular disease mortality is getting reduced, vascular endothelial dysfunction and regulating lipid metabolism is also reduced. So diabetic patient also, polyphenol is a very good, improved glucose control and insulin sensitivity. There is an amazing thing. So polyphenol, as I told you, the double-sworded kind of double-edged sword so far as antioxidant property is concerned. Is a very efficient, particularly the flavonoids, flavones, phenolic acids, etc. Then the question, obviously, why plant source of antioxidants are important? Why people are now in search of plant source of antioxidants? Because the physical properties of synthetic antioxidant like BHT, BHA, which was in vogue for a long time, because of their carcinogenic non-target effect, their use have been banned in the first world countries. Only some process industries, they are using this. Okay, otherwise, since they have the non-target effect, their use is totally banned. Okay, so in view of the increasing risk factors, and this natural source of antioxidants, people are in hunch of the natural source of antioxidants, particularly which are present in medicinal plants are as a dietary supplement for therapeutic antioxidant. So it has been reported that there's an inverse relationship between the dietary intake of the antioxidants and the occurrence of the degenerative disease. Okay, so if you take much more antioxidant rich food, there will be lesser incidence of degenerative disease and occurrence of the aging. Okay, so use of natural antioxidants of the plant origin moreover is extremely cost effective. You can have a very high source of antioxidants, high titer of antioxidant, which can be easily digested and assimilated further, and they can prevent the kind of diseases you don't want to have. And some Indian, I'm just mentioning so that you can know that what Indian medicinal plants are rich in antioxidant properties, particularly based on their antioxidant capacity and the phenolic content. I have given some names. I'm not reading it out. You can go through. There may be 25 plants which exhibit excellent DPPH and ABTS and FRAP property, as well as very high concentration of polyphenolic compounds and other antioxidant attributes like reducing property, hydroxyl radical scavenging property, so on and so forth. So you can do it also, and they are all reported. You can see that acacia, agel, allium, amaranth, and then aloe vera, all these things are very important. Okay, and if you can keep them in your diet or you can supplement, obviously you'll be able to deter your aging and prevent degenerative diseases. And nowadays you have a functional food concept and a nutraceutical properties. Having nutraceutical properties, then what is functional food in fact? Because the people are after preventive medicine. It is always better if you can prevent a disease at source rather than combating a disease after. So that is the philosophy, and researchers demonstrated that the nutrition plays crucial role in prevention of the chronic degenerative diseases. And the functional food and nutraceutical entered in this concept for considering food not only necessary for living, but also as a source of agent necessary for prevention and the reduction of the degenerative disease. Okay, so they are the agents which can be satisfactorily demonstrated as a functional food to the effect beneficially one or more target function of the body, and beyond adequate nutritional effect and reduction of the risk of diseases. That is, apart from their, what is a nutrient? Nutrient is something which can provide you the carbon skeleton or the metabolic energy. That's it. But apart from that, these are the substances which can put you on an advantageous position by combating your degenerative processes. Okay, that is, they are not allowing the risk of having degenerative diseases. That means, to be very precise, here the functional foods having nutraceutical properties can benefit one or more target functions in the body beyond your nutritional benefits. That is, and these targets are antioxidative effect. They are antioxidative effect. Either they prevent, just like that. Now you think of it that you have a functional food which is having a high concentration of flavonoids. Now the flavonoids are working, and it's not giving you the energy. It's not giving you the architectural compound light. Okay, fine. Or it can be metabolized to give you the carbon skeleton, which can provide you some kind of energy. That's done. Or it can be utilized in some other metabolic processes through its incorporation. That's done. But on top of these, what is happening here is that you see flavonoid, they can prevent genesis of reactive oxygen species. Suppose your cell is exposed to xenobiotics, and the xenobiotics got the every character to expedite the formation of reactive oxygen species and oxidative damage. So that can be prevented at source. And not only that, if at all they have generated ROS and your cell is exposed, already experiencing an oxidative stress, they, through their chain breaking reaction, they can regulate the redox homeostasis and bring it back so that the healthy status can be resumed. So that's the thing. And the plant source rich in antioxidants always reduce your chances of having degenerative disease. Now, so whatever I told you, I just like to sum up, because I've taken much more time. I believe maybe you got bored. And but now let me tell you that antioxidants, if I have to sum up whatever I have told you, antioxidants are important component. It's not a simply agent which can interact with ROS, detoxify them. No, don't put it in that way. After this, you should comfortably say they are the agent of an important, rather they are an important component of an elaborate redox system in all aerobic cell. And the redox systems are ROS, antioxidants, ROS sensors, ROS signaling cascades, ROS induced your redox regulated genes, so on and so forth. So it's an important component. And what will be the fate of the, or rather oxidative fate of the cell in an aerobic environment, which we cannot avoid, because we are using oxygen for our own benefit. So we have to deal with this particular molecule very, very cautiously, and that's why these entries are there, and they are not only to detoxify them, but they want to keep them under tight vigil, and they want to keep them under regulated concentration, and so that antioxidant is working more as a redox buffer, where they regulate the endogenous concentration of reactive oxygen species to such a level where it can evoke its beneficial role, not an oxidizing your catastrophic role. Okay, so the antioxidant, ROS antioxidant interaction at metabolic interface actually determine the fate of the cell. That what will be the fate of the cell, whether it is going towards deterioration or oxidative threat, or it is going towards redox signaling and defense process. So that's a very important cue. And antioxidant incompetence, if you have antioxidant deficiency, antioxidant incompetence, so on and so forth, that always instigates the oxidative threat and or oxidative stress, and which is not at all good for the cell. And the aerobic cell particularly poses an elaborate antioxidative system based on their chemistry, as well as their recruitment and their orchestrating action inside the cell to control the environmental stress mediated oxidative events. Okay, so that's a very important thing. And plants precisely exhibit an well orchestrated redox regulation mechanism where the antioxidant are regulated. So the function, the kinetics, I haven't shown you the experimental result intentionally, because that would have taken much more time. It's not possible to vouch everything experimentally, whatever I'm claiming, because that would have taken much more, because these are very serious issues. Only suppose this point, plant exhibit well orchestrated redox regulation mechanism to sense and combat environmental odds. It itself is a talk. Okay, so it's not feasible. So that's the point, that redox regulation is a very important avenue. You may be under environmental stress, you may be under degenerative disease. So for example, COVID, these days of COVID, COVID infection is biphasic. For your information, the initial 7 to 8 days, you'll have hardly any complication, but after 8 to 9 days, the complication starts. Why? Because your cytokine storms are initiating after 8 to 9 days, and cytokine storm is an inflammatory response, and your inflammatory response largely depends on the genesis of the reactive oxygen species. So if you have a very strong antioxidant competence, if you through functional food property, you have preventive antioxidant therapy whatsoever that you have to inculcate, then you can probably combat this disease. Okay, so you can see that any kind of disease, degenerative disease, I told you more than 100 degenerative diseases, right from cancer to atherosclerosis, in one way or another, depends on reactive oxygen species mediated inflammatory responses, or as mediators. So they are the mediators, you see, and here the role of antioxidant is very, very important if we have to check it. So you have to have a very organized and well-regulated, orchestrated redox regulation mechanism. Otherwise, you cannot combat this. And antioxidant, as I've already mentioned, that they exhibit diverse kind of defense lines, defense mechanism. 4 lines of defense I have discussed. So please keep it in your mind that it is not merely the chain breaking reaction, but it also quenching transition metals. It also stabilizing antioxidant, working as co-antioxidants, or through redox regulation, through redox regulation and genesis of its own antioxidants, it works. So, and in animal systems like human being, these are the most, the ROS is the most damaging contributors, and particularly under aging or under chronic degenerative disease. So antioxidant therapy is a very important, promising, you can say, field of work in that context. So emerging research evidence suggests that antioxidant can control auto-oxidation by interrupting and propagation of the free radicals, particularly in response to these xenobiotics and other oxidative threat, and improve your immune function and increase the healthy longevity or the healthy status and the longevity of animals like us. So in this backdrop of banning synthetic antioxidants, the search of natural antioxidant is getting importance, and the plant source of antioxidants, particularly as a nutraceutical, is gaining importance, getting momentum. So lastly and not the least, this is my perception that antioxidants are the companion of the aerobic cell since the evolution with diversified chemistry and the works as the versatile molecule, and that influence and regulate biology of cell more as a redox buffer while integrating themselves with the defense signaling associated with stress tolerance, prevention of disease, aging, and other developmental processes. Thank you a lot for your patience hearing, and I'll be happy if you have question. Hope I will.
Thank you, sir, for such a nice, elaborate, and enriching presentation. We have another webinar organized by NSS. So before we move to the question answer session, I would like to invite our Honorable Principal Sir, Dr. Tapu Shamanto, to share his insights with us. Sir, please.
Okay. Are you listening?
Yes, sir.
Okay.
Yes.
Okay.
Yes, sir.
Okay. Very good afternoon. I am very much thankful to today's speaker, Dr. Soumen Bhattacharya, Head and Associate Professor, Department of Botany, Head and Professor, Department of Botany, University of Bardwan. I am also thankful to all the participants who have paid their patience listening here. I am also very much thankful to my students, my teachers, and my fellow colleagues who have come together to success this national level webinar. I will say very few words on today's discussion, and I will discuss in the light of the chemistry people and as a layman's perception, because two important things is here. One is the free radicals, which are related with the aging, and another is the antioxidant. Now, how antioxidant they are reacting or affecting the free radicals, which are very much related with the aging, that will be discussed. And first of all, one important thing is that how and why these free radicals are being generated. This is what is the basic chemistry is there. Now let us, if we see the board here, we can. If you see the board here, is it visualizing?
No, sir.
Is
No,
it
sir.
seeing?
No, sir.
No, sir.
Are you seeing my board?
No, sir.
Okay, there is a problem. What happens? হ্যাং করে গেছে।
Sir, our respected speaker,
Bye-bye.
Moment, sir. Sir, if you just off the your screen presentation, then I think that our principal sir will share his screen.
মেশিনটা হ্যাং করে গেছে, হ্যাং করে গেছে। প্রেজেন্টেশন স্টপ প্রেজেন্টিংটা কাটতে চাইছি, সেটাও কাটছে না। তো এইটা তো প্রেজেন্টেশন দেয়া ছিল, কেটে দিয়ে আবার করতে চাইছি।
Somen Bhattacharya is presenting.
না, ওটা ওর
I think if our respected speaker Somen Bhattacharya sir, if you please stop your presentation, then I think our principal sir can present. Thank you, sir. Principal sir, I think now you can present.
কোনোটাই হচ্ছে না, কথা হচ্ছে না কেন? অনেকগুলো খুলে গেছে।
না 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 না, অনেকগুলো খুলে নেই।
এই এই এই।
ওর স্ক্রিনটাই হয়েছে।
হ্যাঁ,
না না, ওইখানে না,
ওর
এটা
স্ক্রিনটা
তো ওর স্ক্রিনটা দেখাচ্ছে না।
ওর স্ক্রিনটার উপরে তো আমার হ্যাং করে গেছে।
নিতে পারছে না, বেশি চাপ পড়ে গেছে।
বেরিয়ে গিয়ে করতে হবে।
কন্ট্রোল ডিলিটও নিচ্ছে না।
Requiring security options.
কি ক্লাস নাকি সেই?
না না, সেই ওয়েবিনার। আরে
আসবে।
এবার মনে হচ্ছে এসছে।
Joydeep, will you excuse me? I will take a leave.
Sir, only there are 2 questions. So let's do the question answer session. Riddhi ma'am, please pass the 2 questions.
Okay, sir.
Please.
Our first question from Dipan Banerjee. How does the cell prevent oxidative damage by producing ROS? And how does ROS cause oxidative stress in plant body?
So this is very simple. I have told it. Maybe he skipped. Oxidative stress is a situation where the redox homeostasis of the cell is disturbed due to the overactivation of ROS or incompetence of antioxidant defense. So your kinetics of generation of ROS is more compared to the efficiency of antioxidants that used to work in the metabolic interface. And if it is so, then you have an oxidative stress. But again, oxidative stress can be divided into 2 types. One is sublethal stress, where the oxidative stress works as a redox signal, and another oxidative stress, lethal oxidative stress, where the oxidative stress causes oxidative damages. You got my point. So all oxidative stress is not necessarily instigate oxidative damages. You know, if it is sublethal, where the ROS level is being kept to the lower magnitude, it works as a redox signal. So that's beneficial. It itself instigates the defense genes, okay, through its own signaling mechanism. And how antioxidant deter it? I have given the 4 mechanism of action, 4 lines of action. You know, just go through this. By chain breaking, by sequestering with the transition metal ions, by working as an antioxidant coupled redox regulation, and by working as co-antioxidants or an antioxidant stabilizer, they are working. Second question, another question?
Yes, sir. Next question from Chandan Mollik. Can antioxidant itself induce oxidative stress at higher dose?
No, if there's a very tricky question, you know, if you since reactive oxygen species is not always harmful, if you give reactive oxygen species under tight regulation in a low concentration, then it can work as a signaling agent and evoke defense signaling. So if you put high concentration of antioxidant that causes the 10% detoxification or rather 10% reduction of your reactive oxygen species, so there is no remnant reactive oxygen species left to the system, then it is harmful because your defense mechanism, defense signaling through redox regulation or through ROS signaling is not operating. So this is one of the important drawback of antioxidant administration without knowing the internal details of antioxidant status.
Okay, sir. Next question from Nivedita De. Sir, is there any optimization of accumulated ROS amount or of accumulated ROS after which further accumulation can be considered as major tilt in AOX ROS balance?
This is something which depends on cell type to cell type, tissue type to tissue type. This is not universal thing, and not only that, it depends on the developmental stage also. So in which developmental stage you are going through and whether you are under stress or under ambient environmental condition, this has to be taken into consideration. Suppose you are under stress, so you should have one kind of requirement. Suppose you are under ambient environmental condition, so you should have another kind of environment, the regulation. So it cannot be equated. It depends on cell to cell type, organ type, and the developmental phases through which it is passing through, or whether they are imposed to oxidative or environmental threats or not. All these things, depending on all these things, you have to assess it. You know, and you can assess it easily that whether there are some indices. That indices are where if you find that the oxidative damage parameters are getting enhanced, whereas the MAF kinase activity or the transcript expression is getting lesser. So you can say that, well, now it is something which is moving towards the major tilt, and it is devastating. But if you see that there is a transcript expression of the defense genes,
and there is less accumulation of oxidative products like carbonyl contains or other products. So we can say that well, there is a minor tilt and of under this situation.
Okay, sir. Another question from our faculty. Production of ROS is one of the earliest cellular responses following pathogen recognition and are important signals mediating defense gene activation in plants. Does antioxidant hinder this process and act as a necessary event?
No. Rather, antioxidant facilitates this process. Why I'm saying this? So when an elicitor comes in contact with the host, there is a decontrolled generation of reactive oxygen species. Now it is antioxidant which buffers the concentration of ROS to that extent which can instigate a redox signal. You try to understand, because when an elicitor comes in contact with the host, there are plenty of NADPH oxidase got activated, and there is a burst of ROS formation. Now these ROS, if you keep it as such, they are going to affect so far as oxidative damage is concerned. Now, if there lies the significance of the antioxidant which buffers the endogenous concentration of ROS and keep it in a range which is rather signaling in nature, because so if you have to induce a signaling agent, the endogenous concentration of ROS has to be regulated. So this regulation is being done by antioxidant. So antioxidant is not harming. Rather, it is helping you out all the way to produce an endogenous ROS signal in terms of a specific titer of ROS, which can induce signal. That's it.
Now, I would like to request Professor Dr. Joydeep Acharya sir to deliver the vote of thanks and formally conclude the session. Professor Acharya sir, please.
Thank you so much, sir. Thank you so much, sir, for such an enriching session. We all are extremely grateful that you could take time out from your busy schedule to address the participants here. I can firmly say that we all have benefited from exercise. I'd like to apologize because there's some sort of technical hindrance in this platform. That took a little time to start our program, and our principal sir is also in the queue to deliver his short motivating speech. Thank you very much, sir.
Thank you. I'll take a leave. Thank you, everybody. And one just humble correction. I'm not head of the department presently. I'm just coordinator of Center for Advanced Study. Thank you.
Okay, sir. Okay. So I would now request our participants to please wait for our principal, sir. He is so much cooperative and punctual that he decided to deliver his enthusiastic speech after Professor. So our principal sir has joined. Now I would like to request our sir, our respected sir, to deliver his lecture.
Okay. What I was telling then at that time, that two important things is here regarding our today's topic, severe antioxidants. Although our society nowadays recently called these things severe oxygen, because in this pandemic time, this is the need of the day. Whatever it may be, but I was telling, if we understand these topics in the angle of the chemistry and from the chemical point of view, if we want to see our discussion slide through presentation mode. Now it is saying, it is visualizing.
Yes, sir.
Okay. This is antioxidant and free radicals. I have told you earlier that these are the two important things. One is the antioxidant, another is the free radicals. So there are so many chemicals and natural products which have the ability to scavenge the free radicals. So many things are there which can scavenge the free radicals or oxygen, free oxygen radicals in the body or in the in vitro systems. So these things, they are called the antioxidants. And another thing which are also important is called the free radicals, and how these free radicals, they are being generated during the course of reactions. In chemical reactions, what we have known regarding a chemical reactions, if we see a chemical reactions like this, suppose a reactant A is giving you the product B and C. So what is our common chemical consequence? The product reactant A, it is just being divided into two products B and C. But actually it never happens. In chemistry, almost in many cases, these are not happen. A react with another A molecules giving you the B and C. This is generally not happens. Suppose a reaction like this.
Lockdown extend for you.
Hello? Are you listening?
Yes, sir.
Okay.
Yes, sir.
Okay.
Sir, your border is not visible.
Sir, your screen is not visible. Yes, sir.
Screen is not visible.
Yes, sir.
No, sir.
That you are presenting not visible.
Why? Okay. I am trying. আমার ল্যাপটপটা একটা প্রবলেম করছে। It's maybe the network problem or maybe the laptop problem, but it is creating problem. I am trying to show you. Is it now visualizing? No, it is not visualizing. Still.
No, sir.
Why? Okay. I am again trying. I could not know why it is not being presented. I could not know. Okay, I am trying. Is it visualizing?
No, sir.
No.
So it is written that stop presenting to avoid mirroring, don't share your entire screen. Just some sort of writings are there in the screen, sir. But the whiteboard is not visible.
No, it is whiteboard. It is a slide. PowerPoint slide. Is it not visible?
Sir,
The
then
screen
your,
is,
Sir. Sir, your screen is visible, but now please open the PowerPoint slide actually. Then it can be visible to us.
Okay. Screen is visible. Is it visible? I think color of the font is very light. That's why it is creating problem.
No, sir.
No, sir. Sir, kindly put your slide in presentation mode.
Your sliding presentation, not in PowerPoint presentation. Is it now visible?
No, sir.
Okay, is it visible?
No, not yet, sir.
Not yet.
No, sir.
Okay, I am telling you with this slide also. I am telling you why it is not visualizing. That could not understand to me, but I am telling you. You are just listening. Now you see free radicals are the reactive intermediates that are generated during the course of any reactions. If you see, suppose A react, A is the molecule which is reacting to give you the product B and C. That means it does not mean that two A molecules, they are reacting to give you the B and C. আমার কথা শোনা যাচ্ছে তো?
একদম
Yes, sir.
শোনা যাচ্ছে।
আমার কথা শোনা যাচ্ছে। Okay. Now if you see, I could not understand why it is not visualizing to you. Now if we consider the oxidation of acetaldehyde. So oxidation of acetaldehyde, it gives you the methane and carbon monoxide. But apparently it looks like a first order reaction. That means this acetaldehyde, the concentration, first power of the concentration of the acetaldehyde, it depends upon the rate of the particular reaction. But actual mechanism shows that this is a reaction of 3/2 order reaction. That means 1.5 order reaction. And how and why this half order reaction comes, 1.5 order reaction comes instead first order reaction, that was the question. And after finding, it was found that there is a different type of mechanism. Actually, not a single acetaldehyde molecule, they reacting with each other gives us the methane and carbon monoxide. Instead of that, a acetaldehyde molecule, it first generate a methyl free radical along with the aldehyde radical. And this methyl free radical, these are actually the reactive intermediates. Actually, almost in many cases, or in most cases, the reactions occurred through some reactive intermediates, where the reactive intermediates are the free radicals. And this is the genesis of the free radical, which is being generated during a course of any reactions. And that's why free radicals are important in case of what was discussed here, antioxidant and free radical. So what is the genesis of the formation of free radicals? So here the methyl free radical is being generated from the aldehyde, acetaldehyde molecule, and this methyl free radical, it is again react with the another molecule of acetaldehyde and will give us a methane and CH3 CO radical. And this CH3 CO radical again gives us another methyl radical and carbon monoxide. These CH3 CO radical and these CH3 methyl radical, these are actually the reactive intermediates, and these are free radical which are being generated during the course of the reaction. And almost in many chemical reactions and almost in our life process, in plant life process, there are so many reactions where these free radicals are being generated during the course of reaction, and those radicals, those are called the reactive intermediates. And these reactive intermediates, they are very significant. They are very significant to propagate a reaction. And these reactive intermediates, the basic nature of the reactive intermediates are that they generated during the course of the reaction, and they have been used during the course of the reaction. That's why these free radicals or free radicals are the reactive intermediates, and they are being generated during the course of the reaction and form the ultimate product. That's why in our discussion, free radicals and free radicals and scavenging materials, these are the two important things. One is the free radicals. These free radicals are being generated with the progress of our age. That means free radicals are associated with the aging of the persons. Similarly, some materials, some chemicals, some natural products, they have the ability to destroy or to consume these free radicals. I shall tell you only a single chemicals which is very much important in chemistry, because in complex chemistry, because it is important in complex chemistry, that's why I shall discuss or I shall tell you some information, few information about that chemicals or that natural products, which is called the flavonoids. These flavonoids, this is a very good scavenging materials. Perhaps all of you know that these flavonoids, it contains basically the polyphenols. There are so many polyphenols, you know, there's catechol, resorcinol, guaiacol, all these things, they are polyphenols, and these basically flavonoids contains the polyphenols compound. And in plant, wherever there is a pigment which form the color, that means most flower, fruit, seeds, etc., they contain the flavonoids. আমার বোর্ড কি এখনো দেখা যায়নি?
No sir.
No sir. Okay. Flavonoids is generally attributed to their antioxidant property. We have listened those things. Our honorable speaker elaborately discussed about the how flavonoids are participated in the oxidative reaction in plant system or body system. Now, hello. আমার এটা কাজ করছে না গো। আচ্ছা, আমার কাছে স্টপ প্রেজেন্টিং আছে আর হচ্ছে ইগনোর নেই। আচ্ছা, হাইড আছে, হাইড করলে তো আমারটাই চলে যাবে। গুগল মিটে যে স্ক্রিনটা আছে, এক সেকেন্ড আই এম গোয়িং। গুগল মিটে যে স্ক্রিনটা আছে, হ্যাঁ, স্টপ প্রেজেন্টিং। নো ইগনোর ইজ দেয়ার। কোথায় তুমি দেখতে পাচ্ছ, কোনখানে বলো? স্টপ। ওকে। Is it visualizing?
Yes sir. Now yes. It's better, I think. Yeah.
Achha, okay.
No sir, the screen is not over here.
It's a PowerPoint. It is a PowerPoint.
তাহলে স্যার উইন্ডোজ ইয়ে করে পাওয়ার পয়েন্টটা সিলেক্ট করে শেয়ার করুন।
শেয়ার করি সিলেক্ট করছি। হ্যাঁ, আবার স্টপ প্রেজেন্টিং করে আবার করব। বেশ, দুবার করলাম, অভিষেক হচ্ছে না। আমার ল্যাপটপের বয়স হয়ে গেছে। আচ্ছা তুমি বলো, let you speak, instruct me how to do. প্রেজেন্ট আমি তো করেছিলাম। আচ্ছা, আমি আমার এটাকে বন্ধ করি। এটা বন্ধ করবো তো নাকি না? ভিডিও অফ করে দেবো। ওকে, প্রেজেন্ট নাও করছি। এন্টায়ার স্ক্রিন দেবো না উইন্ডো দেবো? উইন্ডো দিলাম। উইন্ডো দিয়েছি। আচ্ছা, শেয়ার করব। হ্যাঁ, সিলেক্ট করেছি। সিলেক্ট করে তো শেয়ার করতে হবে, তাই তো? তাছাড়া
হ্যাঁ স্যার।
হ্যাঁ হ্যাঁ হ্যাঁ, প্রেজেন্টেশনটা সিলেক্ট করবো আগে।
হ্যাঁ
হ্যাঁ হ্যাঁ হ্যাঁ হ্যাঁ হ্যাঁ হ্যাঁ হ্যাঁ, আমি শেয়ার করছি।
এবার হবে।
আরে এটা তো প্রত্যেকবারই হলো। হয়েছে?
না
হয়নি তো?
স্যার স্যার একটা কথা বলবো,
হ্যাঁ,
আপনি
বলুন।
একটু মিনিমাইজ করুন। আপনি এই যে শেয়ার করেছেন, তো আপনি একটু
হ্যাঁ, হ্যাঁ।
মিনিমাইজ করে আপনার যে ফাইলটা আপনি শো করতে চাইছেন ওটা আপনি একটু ওপেন করুন, দেখুন খোলে কিনা। আপনি পুরোটাকে মিনিমাইজ করে দিন, এই অবস্থাতেই মিনিমাইজ করুন।
হ্যাঁ, মিনিমাইজ তো
Minimize
করেছি।
আহ,
না, হোল
Okay.
উইন্ডোতে প্রেজেন্ট করলে স্যার আপনাকে গুগল মিটটাকে মিনিমাইজ করতে হবে।
হ্যাঁ, না পাঠানোর কি আছে?
হ্যাঁ, আমি সেটা করতে পারি।
আচ্ছা আচ্ছা। ওকে ওকে ওকে, I am sending you. ওকে।
আচ্ছা।
বাবা, আমাকে তো এখান থেকে বেরিয়ে যেতে হবে গো, নাকি?
না না, বেরোতে হবে না।
আচ্ছা আচ্ছা আচ্ছা, বুঝতে পেরেছি, আমাকে আর একটা খুলে নিতে হবে আর কি। বেশ। হ্যাঁ,
স্যার হচ্ছে স্যার
actually
এন্টার
minimize
স্ক্রিন
করে
সিলেক্ট হয়ে
খুললে
গেছে।
কিন্তু দেখা যেত, মানে মিনিমাইজ
আচ্ছা, কেন বলো তো? আচ্ছা, কেন কেন তাহলে দেখো, এখন দেখা যাচ্ছে?
স্যার আপনি একটু মিনিমাইজ করুন না, উপরে যেটা করা আছে
মিনিমাইজ করেছি।
না,
করুন স্যার।
হয়নি স্যার।
হ্যাঁ,
পুরো গুগল
এখানে দেখাচ্ছে না মিনিমাইজ।
ক্রোমটাকে মিনিমাইজ করুন।
ওপরে একদম ওপরে দেখুন স্যার একটা।
আচ্ছা, উইন্ডোটাকে মিনিমাইজ করতে
হ্যাঁ
হবে।
হ্যাঁ হ্যাঁ,
করলাম, করেছি।
স্যার ওটা ম্যাক্সিমাইজ হয়েছে, মিনিমাইজ না।
স্যার মিনিমাইজটা
আমি
হচ্ছে
দেখতে
মাইনাস
পাচ্ছি।
সাইনটা, একদম ওপরে মাইনাস সাইনটা, যেটা আপনি চৌকো বক্স তার
হ্যাঁ, করেছি করেছি।
এই পাশেরটা স্যার।
হ্যাঁ,
হ্যাঁ
ওটা।
হ্যাঁ ওইটা হ্যাঁ।
এটা তো চলে গেল।
এবার আপনি আপনার প্রেজেন্টেশন ফাইলটা ম্যাক্সিমাইজ।
হ্যাঁ, দেখা যাচ্ছে?
সাদা
হোয়াইট দেখা যাচ্ছে।
না, বলছি দেখা যাচ্ছে না, কিছু একটা সমস্যা হয়েছে।
আমি পাঠিয়ে দিন স্যার।
আরে এ তো করতে করতে পরেরটাও গন্ডগোল হয়ে যাবে। আচ্ছা, এক সেকেন্ড।
হ্যাঁ স্যার, স্টুডেন্টরা আছে তো, থাকবে, অসুবিধা নেই।
আরে স্টুডেন্ট থাকার সঙ্গে কিছু এসে যায় না। দাঁড়াও, আমি এটা থেকে বাইরে বেরোই। আমার একাধিক খুলে গেছে আসলে। এবার মেশিনটা আবার হ্যাং করবে, বুঝতে পেরেছো? স্টপ প্রেজেন্টেশন করে আমি এখান থেকে একবার লিভ করে যাই। দিচ্ছি, আমি প্রসূনকে দিচ্ছি।
হ্যাঁ স্যার।
I think there is a technical problem in the device of our principal sir, as because he informed me just sometimes back that his laptop was hanged. Before the entry of our principal sir, hello, am I audible?
Yes, yes.
Yes, sir.
Yes, sir.
Okay. Before the entry of our principal sir, I would also like to thank our IQAC coordinator, Professor Devasis Bhattacharya, and the members of the advisory committee for their encouragement, especially Dr. Abhishek Mukherjee, from the Department of Mathematics for his technical support. I would also like to thank the member of the organizing committee, Dr. Somnath Mondol, Riddhi Madam, Dipti Madam, Omita Madam, Sushmita Madam, Chandan Sir, Regina Madam. I will also extend my special thanks to Dr. Prasun Biswas, the HOD of our department, as well as the joint convener of this webinar for his efficient technical guidance and constant support. Let's wait for our principal sir for a while. The participants are also very much thankful. I'm going to tell you at the last. I would also like to apologize to the participants regarding the technical issue.
Yes, আমি জয়েন করেছি।
স্যার আসুন স্যার।
প্রসূনকে প্রেজেন্ট করতে বলো, প্রসূনকে পাঠিয়েও দিয়েছি।
প্রসূন স্যার, you please present the screen.
Just give me 1 minute.
আচ্ছা, ওকে। আমার ল্যাপটপটা আজকে খুব ঝামেলা করছে, কি হলো কে জানে।
আমি ওটাই বলছিলাম স্যার, মানে টেকনিক্যাল ইস্যুর জন্য
it has become overburden. একটা সময় হয়ে গেল।
Actually, you are overburdened with work, sir.
That is true.
Yeah, that I am saying for the last 1.5 years.
I, along with my laptop, both have become overburdened. Okay, it is visualizing. Is it visualizing?
Yes, sir.
Yes, sir.
Yes, sir.
Now, আমার কাছ থেকে তো হবে না। Yes. That was I was telling. But the next slide, please. Prasun. Okay. Here you see that the acetaldehyde, it is giving us the methyl radical as well as aldehyde radical. That means free radicals are being generated, not the single aldehyde molecule that is actually giving us this methane and carbon monoxide. Not this is true, but actually it is being generated the methyl radical as well as CH3CO radical. This methyl radical and CH3CO radical, these are called the free radical, and these free radicals, they are actually the reactive intermediates which are being generated during the course of the reaction, and they are consumed with the progress of the reaction. In any chemical reactions, you will see there are some reactive intermediates, and there are some reactions which are called the propagation reaction. That means actually entire reaction is being propagated by 2 or 3 other different chemical reaction and ultimately giving us the product. In life system, in plant system, there are so many reactions, there are so many processes where these free radicals are being generated during the course of the reaction. That's why free radicals are inevitable with our aging process. That is important. Two things I am discussing in our discussion savior of antioxidants. One is the antioxidant, another is the free radicals. So I am telling why the free radicals are inevitable during the course of reaction, during the course of the life processes. Okay, now next slide, please. Now there are so many chemicals, so many plant products, so many natural products which have the pigments. That's why which makes the color of the flower, color of the fruits, color of seeds, all these things. These are basically for the flavonoid compounds. These flavonoid compounds are nothing but the polyphenols. Polyphenols are like you know that. Phenols, you know, but polyphenols are catechol, resorcinol, guaiacol. So many polyphenols are there, and these flavonoid compounds, they are very much responsible because these flavonoids compounds can be complexed with the transition metal elements, and that's why they are important. These flavonoids, it is generally attributed to their antioxidant properties. So why flavonoids produces their antioxidant properties? Because they can consume the free radicals which are being generated with the course of the reaction, with the course of the life process, with the course of the body system, course of the plant body system. These free radicals are being generated. If you see here, the free radicals like oxygen free radical, hydroxyl free radical, this is nitric oxide free radical, this is O-methyl free radical, this is carboxylate ion free radical. So all these free radicals are being generated during the course of the life process, and these free radicals can be consumed with the by the flavonoids, which are basically the polyphenol compounds. Now the flavonoids may inhibit one important thing that was also discussed by our guest speaker. These flavonoids, it can inhibit the generation of primary oxygen radical. That is important. Oxygen radical through the life process, আমাদের কাছে তো দুটো জিনিস ছিল, একটা হচ্ছে ফ্রি র্যাডিক্যাল তৈরি হচ্ছে কেন, আর আরেকটা হচ্ছে এই ফ্রি র্যাডিক্যালটাকে কনজিউম করতে পারে কোন কোন কেমিক্যালস বা প্ল্যান্ট প্রোডাক্টস, বা আমাদের বলা যেতে পারে ডায়েটারি কম্পাউন্ডস, কোন কোন প্ল্যান্ট প্রোডাক্টস এবং ডায়েটারি কম্পাউন্ডস যেগুলো এই এজিং প্রসেসে যে ফ্রি র্যাডিক্যালস তৈরি হচ্ছে সেগুলোকে কনজিউম করতে পারে। Now the flavonoids, why flavonoids are important here? Because flavonoids, they can inhibit the generation of the primary oxygen radical. These primary oxygen radicals which are being generated during the course of the life process, they can be consumed. They can be consumed by the flavonoid compounds. The flavonoids are basically the plant material, plant polyphenol materials. They can consume these primary oxygen radical. This is important. This primary oxygen radical that can be consumed by chain oxidation or by forming the transition metal complex of the flavonoid compound. That's why these flavonoids are called the chelating materials, because they form the metal chelate compound or metal chelate complex. Actually, they form the metal chelate complexes, and these metal chelate complexes, they can destroy this oxygen free radical which is being generated during the course of the reaction. Now you see here this rutin, taxifolene, these are polyphenols. These are different type of polyphenol, have the higher scavenging potencies towards the superoxide. That was telling also by our speaker, the superoxide that can be scavenging with the help of the metal complexed polyphenols or metal complexed flavonoids. By using the flavonoid metal complexed, one can easily scavenging the superoxide which is also being generated during the course of the life process or during the course of the life system or plant life system. Now next slide please. Next slide please. Okay. Flavonoids chemical properties. You see, complex flavonoids are oxidized significantly lower than the free flavonoids. That's why complexation of the flavonoids are important. And these complexation is possible in case of copper, in case of ferrous and ferric complex. That can be occurred, and that can be happened. The metal iron complex flavonoid or ferric iron complex flavonoids or copper flavonoid complex, they are significantly lower than the free flavonoid oxidizing ability. Flavonoid complexed metal is most active antioxidative center. If you form the metal complex flavonoid, that means that metal which is being complexed with the flavonoid, it forms a very active and antioxidative center. তার মানে কি তোমার আমার প্ল্যান্ট সিস্টেমে কিংবা বডি সিস্টেমে যে ফ্ল্যাভোনয়েড রয়েছে সেখানে যদি কোনোভাবে আমরা আয়রন অথবা কপারের মেটাল কমপ্লেক্স, কপারের তে অসুবিধা আছে কিন্তু মানুষের বডি সিস্টেমের কথা যদি আমরা ভাবি সেখানে যদি আমরা আয়রনের কমপ্লেক্স তৈরি করতে পারি, তো সেই আয়রন কমপ্লেক্সটা that complexed metal should be the very most effective antioxidative center which can easily scavenge the free radicals which are being generated by the life process. তার মানে এইভাবে আমরা যদি কোনো একটা প্ল্যান্ট মেটেরিয়াল যেগুলো প্ল্যান্ট ফ্ল্যাভোনয়েডস বলা হচ্ছে সেগুলোকে যদি আয়রনের কমপ্লেক্স তৈরি করতে পারি বা ট্রানজিশন মেটাল কমপ্লেক্স তৈরি করতে পারি তাহলে সেই কমপ্লেক্সড ফ্ল্যাভোনয়েডটা সেটা হচ্ছে that would be the most active antioxidative center which can destroy or which can destruct the free radicals which are being generated during the course of our life. Now complex flavonoid with iron to oxygen and hydrogen, that is important. Complex flavonoid আমরা যেটা বলতে চাইছি যে বডির মধ্যে যদি আমরা কমপ্লেক্স ফ্ল্যাভোনয়েড তৈরি হয় কোনোভাবে by using some material, by using some drugs or like that, by using some chemicals, if complex flavonoids are being generated within the plant system or within the body system, then it will decompose the superoxide. Superoxide means O3 radical. So that superoxide can be decomposed with the help of the complex flavonoid. That's why complexation of flavonoid by metal, it is also a very nice chemistry, and by using or by applying that chemistry one can prevent the oxidative degradation in the life process. And the last thing is that metal chelation process promote the penetration of the flavonoid in the hydrophobic region of the biological membrane. You know that biological membrane which contains the unsaturated carbohydrate, and they are to some extent hydrophobic in nature. Unsaturated hydrocarbon which are present in the biological membrane, they are to some extent hydrophobic. That's why some materials or some chemicals cannot be penetrate into that into those areas, through those areas. But if you form the metal complex or metal chelation of flavonoid by iron or something like these transition metals, these metal complex, they can penetrate through that unsaturated hydrocarbon hydrophobic zone of the biological membrane, and in that way it can also penetrate the biological cell by forming the flavonoid metal complex. And lastly, the complex flavonoid are oxidized significantly lower than the free flavonoid. Free flavonoid যতটা না অক্সিডাইজ করতে পারবে ফ্রি র্যাডিক্যালগুলোকে কনজিউম করতে পারবে তার থেকে কমপ্লেক্স ফ্ল্যাভোনয়েড, it has been found that complex flavonoid can easily oxidize the free radicals or oxidative radicals than that of the free flavonoids. That's why complexation of the flavonoids is more significant. Similarly, flavonoid complexed metal is most active antioxidative center, I have told you earlier. Now the complexes of flavonoid with iron and copper can decompose superoxide radical and oxygen and hydrogen. So it can easily decompose the superoxide radical to form the oxygen and hydrogen. So that is another important thing. I have told you earlier that unsaturated double bond case and the metal chelation process. Metal chelation may promote the penetration of the flavonoid. I have told you earlier. Next slide, please. Next slide. Yes, it is. Now this metal complex. See here, this metal is nothing but the pure metal. It is a metal complex, metal flavonoid complex, and this metal flavonoid complex, it's react with the oxygen free radical. This is O2 radical. This O2 radical is react with this metal complex and giving you the metal ion as well as oxygen. So this complex metal is reacting with the oxygen radical, oxygen free radical, and giving us the metal complex because they have the very low redox potential, as because these flavonoid complex have the very low redox potential. That's why these type of reactions are being performed within the cell system or within the life system or within the plant or animal life system is possible. Similarly, this metal complex, this complex metal, it is react with the O2 negative ion and hydrogen ion again gives us the hydrogen peroxide, because this hydrogen peroxide, it is also a significant chemicals for oxidative degradation, degradation of the several processes in the body, plant or animal body system. Now the complex flavonoid prevent the production of reactive hydroxyl radicals through Fenton reactions. You know the Fenton reagent. Fenton reagent is nothing but the ferrous sulfate and hydrogen peroxide, and this Fenton reagent is being used as effluent as a chemicals in the effluent treatment plant of several industry. But this Fenton type of reaction has become stopped due to the complex flavonoid. Complexation of the flavonoid, it prevent the Fenton's reaction, because in Fenton's reaction, the hydrogen peroxide, it is generated the several hydroxyl OOH radicals, hydroxyl radical, OOH radical. These are generated through the Fenton's reaction. If there is some flavonoid metal complexes, these flavonoid metal complexes can stop or can prevent the Fenton's type of reaction. And why it is essential, why it is important? এটা ইম্পর্টেন্ট কেন? কারণ হাইড্রোজেন পারঅক্সাইড বডিতে থাকলে সে আরো নানা ধরনের বিক্রিয়া করে ওই হাইড্রক্সিল, অক্সিজেন এই সমস্ত রেডিক্যালগুলোকে জেনারেট করতে থাকবে। বাট this complex flavonoid, they react with the hydrogen peroxide, which is being generated through the cell reaction, different cell reactions, different reactions which are being generated through the DNA, RNA systems. These hydrogen peroxide, which are being generated through the life processes, they are being react with the flavonoid metal complexes, and there is a chance to stop the Fenton type of reaction. That's why this flavonoid metal complexes, it is also important for scavenging the free radicals in our body system. I shall conclude. I shall conclude here by saying 2 or 3 important words. One is the we have to be understood why free radical generation is essential. How and why free radical generation is essential. সেটা আমরা বুঝতে পারলাম যে কেমিক্যাল রিঅ্যাকশনই বলি আর বায়োলজিক্যাল রিঅ্যাকশনই বলি, most of the reactions they are going through the free radical mechanism. That's why free radicals will be generated, and these free radical generation of the free radicals, it is very much correlated with the aging of our system or aging of our life system. That's why free radicals will be generated. Now another thing, so we have to be managed. We have to be managed how these free radicals which are being generated in the course of the reaction be consumed by our dietary system or by our life habit. That was actually discussed on today. এইটাই আজকে কিন্তু আলোচনা হলো মুখ্যত, যে আমাদের লাইফ প্রসেসে যে সমস্ত কেমিক্যাল রিয়াকশন তৈরি হয় বা আমাদের মেটাবলিক সিস্টেমের সঙ্গে কিংবা আমাদের লাইফ প্র্যাকটিসের সঙ্গে যে সমস্ত কেমিক্যাল রিয়াকশনস তৈরি হয়, সেখানে জেনারেশন অফ দি ফ্রি রেডিক্যাল it is inevitable, but the scavenging of this free radical, it is necessary. আমাদের প্রয়োজন to scavenge the free radicals, এবং সেক্ষেত্রে আমাদের কি ডায়েট হওয়া উচিত, আমাদের কি হ্যাবিট হওয়া উচিত, বা কোন কোন জায়গায় কোন কোন কেমিক্যালস, কোন কোন মেটেরিয়ালস এইগুলোকে প্রিভেন্ট করতে পারে, প্রোটেক্ট করতে পারে বাই সাম কেমিক্যাল রিয়াকশনস, বাই সাম বায়োলজিক্যাল রিয়াকশনস, এবং কোন কোন জায়গায় আপডেটেড রিসার্চ পসিবিলিটিস রয়েছে, সেইগুলো কিন্তু আজকে মুখ্যত আলোচনা হলো। আমার মনে হয় আজকে আমাদের স্টুডেন্ট যারা ছিল এবং আমাদের ফেলো পার্টিসিপেন্ট যারা ছিলেন, তারা আমাদের স্পিকার, আমার ফার্স্ট হাফে প্রথমত আজ সারাদিনই আমার ল্যাপটপটা খুবই সমস্যা তৈরি করছে। তার কারণ হচ্ছে ল্যাপটপটা মাঝে মাঝে হ্যাং হয়ে যাচ্ছে, কারণ এত স্টুডেন্টদের রেজাল্ট এবং তাদের ওই ফর্ম ফিল আপের ফাইল আমার ল্যাপটপে হাজির হয়েছে। আমি প্রসেস করে ওবেলায় শুরু করতে পারলাম না, সেই জন্য আমি 10 মিনিট সময় চেয়ে নিলাম, কিন্তু সেই সমস্যাটা আমার রয়েই গেল। আমি জানি না নেক্সট এর সময় আমি কিভাবে করব। এনিওয়ে, তো ইম্পর্টেন্ট থিং ইজ দ্যাট যে আমাদের কাছে দুটো জিনিস ক্লিয়ার হয়েছে আশা করি। সেটা হচ্ছে এই অ্যান্টিঅক্সিডেন্ট এর যে স্ক্যাভেঞ্জিং অ্যাক্টিভিটিগুলো রয়েছে, এগুলো কিন্তু সব নানা রকমের কি বলবো ফ্রন্টিয়ার লাইন অফ রিসার্চ এখানে হচ্ছে। সুতরাং আমাদের যারা কলিগস, যারা এইসব জায়গায় পড়াশোনা করতে চায়, করার আগ্রহ দেখাতে চায়, এখানে খুব ফ্রন্টিয়ার লাইনের রিসার্চের কাজ হচ্ছে। এই যে কেমিস্ট্রির আসপেক্টে যদি ভাবি, সেখানে হচ্ছে এই যে লো রেডক্স পোটেনশিয়ালে বিভিন্ন ধরনের কমপ্লেক্স যারা হচ্ছে এই যে ফ্রি র্যাডিক্যালগুলোর সঙ্গে রিয়াকশন করতে পারে, এই ধরনের রিয়াকশনের উপরে প্রচুর ফ্রন্টিয়ার কেমিক্যাল রিসার্চ হচ্ছে। বায়োলজিক্যাল রিসার্চের কথা তো আমাদের স্পিকার বলেইছেন। সুতরাং আমি আশা করব আজকে যারা এখানে এই সভায় হাজির হয়েছে, মানে এই মিটিংয়ে হাজির হয়েছেন, আমাদের স্টুডেন্টরা ছাড়াও বিভিন্ন কলেজ বা বাইরের যারা টিচার, আমার দেরি হয়ে গেছে অনেকটাই এবং আমার ডিভাইসটা প্রবলেম তৈরি করলো। এটুকুকে যদি আমরা if we neglect these things, these phenomena, principal speech, if we neglect these things, but what is important here, important thing is that we have to be realized through the life processes, through the in vitro and in vivo processes, what are the inevitable phenomena, which are the inevitable phenomena that are to be studied. Those are to be studied, number one. And number two, how those inevitable phenomena can be checked with the help of the chemical process or with the help of the biological process. In biological process, we have 2 or 3 different ways. In biological reactions, there are some genesis. You have to follow the genesis of the reaction. বায়োলজিতে তো যে কোনো রিয়াকশন তুমি হাইপোথেটিক্যালি বলে দিতে পারবে না যে এই রিয়াকশনটা করলে ওটা আটকে দেয়া যাবে। ওই রিয়াকশনটা লাইফ সিস্টেমে, প্ল্যান্ট লাইফ সিস্টেম হতে পারে, অ্যানিমাল লাইফ সিস্টেমে, is there any genesis of that reaction? Is there any possibility of genesis of those reactions? If there is a possibility of the genesis of those reactions, then you can perform those reactions, and with the help of those reaction or with the help of those ways, you can predict how we can restrict the free radical generation with the plant or animal body system. So this is almost my discussion, and I will say that all the participants who have gathered here, they will be benefited, very much benefited. I have seen the several comments there in the comment box. So many people, they have told that it is a very nice discussion, very informative discussion, very elaborate and informative discussion. So I think that all the people who have gathered here, they have been what I will say, they have been inspired with this type of scintillating discussion. With this few words, I shall conclude my talk, কি জয়দীপ আমাকে শেষ করতে হবে নাকি না তোমরা শেষ করবে?
আমি করে দেবো স্যার, আপনি ইয়ে করুন।
আচ্ছা, with these few words I shall conclude my talk and I am giving the microphone over to Joydeep. Thank you all.
Thank you so much, sir, for
Okay.
Thank you so much, sir.
Okay. Thank you.
For describing in very easy way about the basic chemistry behind the free radicals and antioxidants, highlighting the flavonoids. To me, your speech is just like a subtitle of the above speech. I would like.
Thank you so much.
Last but not the least, my heartfelt thanks goes to all the participants who had shown such huge response to make this webinar a total success. Again, I would like to apologize to the participants regarding the technical issue, due to which most of you were unable to join through Google Meet. Thanks to all of you for joining through YouTube streaming. Before closing the webinar, I will again request all the participants to submit their duly filled feedback form sent in the WhatsApp group as well as in the chat box, so that we can issue e-certificates to you at the earliest. Thanks to all of you again. Please stay safe and get vaccinated. Now I am ending this webinar. Thank you very much.
Thank you.
Joydeep, I am leaving.
Yes, sir. You have another webinar there. Thank you, sir.
As I have another.
I will request all the participants to leave. Thank you very much. PV sir, can we leave? Abhishek sir, just stop the YouTube streaming. Abhishek sir, please stop the YouTube streaming.
I think it will automatically stop after end of the meeting.
So sir, taking your permission, can we leave, sir? Prosun sir.
Okay, sir. Thank you for everyone.
Okay.
I think we can stop this meeting now.